bensound.com. bensound.com.
Bom dia a todos, bem-vindos. Eu vou agora falar em inglês, na realidade, broken English, in respect to our guests. So it's a great pleasure to be here today and start this workshop on TMS, very broad in scope, covering many areas from technology to clinical applications. And I think this is a moment to say thank you to many people that have been involved in this project. I first would like to thank the NeuroMat CEPID. NeuroMat is a project that's being funded by FAPESP, the São Paulo State Research Foundation, since 2013. And in terms of translate to English, it's a Research, Innovation, and Dissemination Center. And this center started under the leadership of Professor Antônio Galvez, that unfortunately left us very early in his career. So he passed away exactly 2 years ago. And I would like to dedicate this activity to his memory. He was a very intense person in terms of research and life in general. And I think what we are, let's say, achieving today somehow was the seeds that he planted 12 years ago when he started this project. So there are many people here that were in this project from the very beginning. I can see Claudia and Rocky. I joined the project in a later phase, but I think it's very fair to dedicate this event to his memory. I also would like to thank the FAPESP, of course, that's the São Paulo State Research Foundation. I would like to thank MagVenture, is a company that produces TMS devices, and they are giving us a cough break. This is complicated sometimes to put in our budget, so we have to find this sponsorship. And I also would like to thank the team that put together this workshop. I must say that this was the easiest activity I had organized. That's because I had all the lab group working very intensively, and dedicating many hours to organize. And someone that has organized a meeting know how much time is involved in that, because there are many details that it's not black and white. You have to think and decide, right? So I think we have Thais, Victor Moraes, Carlo, Marco, Deborah, Rebecca, Rakauskas, who else? I always forgot people, so I already so I'd like to apologize if I forgot to mention someone, okay? And also this place, I mean, we are in the center of technology, information technologies, CETIRP, and they are generous offering this space to us, offering all the technical support. So in the name of Marcelo, that's there in the dark room controlling all the transmission, I would like to thank also CETIRP. And of course, this meeting is being transmitted, so this message is also going through the web, and I would like to thank people that are in YouTube watching this event from other places. And I think with that, I would like to stop and turn the word to the co-organizer, Vitor Souza.
Thank you, Bafa, and thank you everyone for coming. Some of us came from quite far. It's a long trip, and it's really nice to see. It's really nice to see so many amazing people together here for us to discuss in the next days. We try to, in our discussions, we try to put together a workshop that has many branches of neuroscience in a way that was all related to TMS, but then we try to put people on the basic research, people from instrumentation development and clinical applications and so on. And the main idea is that we can exchange ideas and experiences, and hopefully we can really create new projects so that we can continue this research. That's the main goal of the event. So please talk as much as you can. That's the only request we want to ask you to do. I will go through a bit of some of the practical things. So because we want to kind of promote discussions and that's something that's really important during the talks. Let's try to, we have reserved more or less 1 hour for each talk. So let's try, the presenter should keep them below 40 minutes so that we have at least 20 minutes for discussions and questions and so on. Leave space for a lot of questions. No need to present all the details at once. So that's one practical matter about the talks. Then we are having this opening session. Then we will have Professor Risto presenting first. We have a coffee break, and then Antonio Roque will present next. Then we have lunch, and then in the afternoon we have 2 more talks and 1 coffee break. We are closing the session at about 5 p.m. So all the days will go more or less about that. Then in the last days we also have presentations with posters and also some oral presentations that have been submitted through the online platform. On the last day, on Thursday, we will have also space for open discussion and a visit to the lab. So one of the driving motivations for organizing the workshop has been that through the collaboration between Professor Bafa's group and Professor Risto's group in Finland, we have established a pretty amazing setup for TMS, which I could say that it's one of the best in the world, in which we combine very advanced TMS technology, multi-channel TMS technology, with robotics and neuronavigation, using multimodal imaging, MRI, and also EEG, EMG. So there's a lot of different modalities combined together to bring technological advance and open new possibilities for clinical applications as well. So the device that we built and the systems that Bafa's team built are all together in the lab now, and we are hopefully going to have a demo session that will work without any major issues in the next days, hopefully, without any big demo effect. Right. And
Victor just mentioned a point that I wanted to highlight, is that as you see, our schedule is not very tight. We are not running from one talk to the other. And we did this on purpose, not because we are lazy. It's that because we want to have more time to interact. And I think it's very important for you to ask questions. I know we Brazilians are kind of shy, and we go to meetings and we listen, we think, but it's when you see the audience from other places, everybody asks. So, in my opinion, there is no stupid question. All questions are relevant. So please, if you have any question from basic to some application, don't hesitate to ask our guests. They are here to do this, to interact, to promote, to spread knowledge. So this is very important, I guess, right? We have like half-hour coffee break. This would be enough for us to socialize. And even after the event, there are other opportunities so you can meet and talk. And the idea is to foster this research area in many more areas than we are doing now at the moment, okay?
Yeah, I think just one thing for those that doesn't have the internet connection, you may use the EduROM if you have like your other institutional connection. But if you don't have the EduROM connection, we are asking for a login to the USPNet Wi-Fi. It should come very soon there. Good timing. Should come very soon there. So reach out to ask for the login for the Wi-Fi. Yeah, I think
this was an example of the efficiency of the working of the organizing team. Okay, I think without further ado, I think we should stop now, and we start with the first talk. Okay, you'll be introducing now Professor Risto Iumoniemi. Professor Risto Iumoniemi is a distinguished professor emeritus of applied physics at the Department of Neuroscience and Biomedical Engineering of Aalto University, Espoo, Finland. In the beginning of his career, he has contributed with several innovations in the area of acquisition signal process from MEG, magnetoencephalogram instruments. And I must say that I first met Risto during the Biomag meetings. I entered this area in less central, 1986 or so, and then we met at the Biomag meetings. And at that time, Risto was a really rising star and still keep rising, okay? So later, Risto developed several transcranial magnetic stimulation techniques, such as navigated TMS, TMS-EEG, and multi-locus TMS. He is the founder and former CEO of Nexteam PLC, the company that brought Navigate TMS and TMS and EEG to the market. And currently, he leads the development of the multi-locus TMS and a hybrid MEG MRI system, and apply these technologies to neuroscience and clinical use. To know a small part of his contributions to the field, we invite you to present this talk. That's the title should be there at the end moment. I don't want to repeat it. And so the podium is yours, Risto, so go ahead.
Let's see if this works. Well, thanks for the introduction and invitation, Buffer, and also the possibility to collaborate. You will see the main result of the collaboration in the lab when you visit the lab here. So I was also given this title. I guess Buffer gave it basic concepts of electromagnetism, TMS, neuronal depolarization and generation of EEG signal. And indeed, please, the most fun thing for me is to have questions. The questions are usually more important than the answers, and simple and critical and stupid questions are the best. What is TMS? So here you see Tony Barker in 1985 to use the TMS stimulator and the coil in his hand over the head of the experimental subject. And this device gives current to the coil, electromagnet, and produces about 1 to 2 Tesla, and in this case, over the motor cortex, and one can then measure.
How can you switch off this connected to VPN?
Oh, disconnected. Oh, actually, I have to connect it. Is it okay? Yeah. Good. Yeah, when you stimulate the motor cortex, you can measure MEPs, motor evoked potentials, from the contralateral hand. So I'll talk about this in more detail in a moment. Tony, in the beginning, they had round coils. So, there's a photograph of a round coil, and this yellow arrow shows the rate of change of the electric current that goes around. There are multiple turns of wire there. And this would be like the surface of the brain below, and it induces similar current. It's like a mirror image, blurred mirror image, rotating in the opposite direction to the rate of change of the current over there. So this is the round coil. And the other coil that is nowadays usually used is the figure-of-8 coil. You can imagine there would be 2 round coils like that, and the current flowing in opposite directions, and then you would have this kind of pattern. What happened now? Is there one area where this electric field, there would be 2 round circles have electric current, and they would add up in the center, current flowing there, so double the current here than in this region, or even more than double. So there's another illustration of the induced electric field with a round coil. You get a circular electric field, angular electric field with a round coil, and more focal electric field with a figure-of-8 coil. So this is sort of the preliminary for my talk. And here you see the wire, what it would look like without the casing, just the wire of a figure-of-8 coil. And you place it close to the brain, over the scalp, and the biggest effect, or the main effect, is under the center of the figure-of-8 coil. The electric current in the brain would go in this figure in the vertical direction. And in practice, there's this casing, and the wires are inside. This is the MagStim coil. Now that we are in Brazil, I visited once the beautiful waterfalls. How do you pronounce Igua's Falls? There's this is also called the butterfly coil, by the way. And I noticed this. I have never seen these beautiful butterflies before, but I noticed it has this 8. So it is actually this figure-of-8 coil. Butterfly is the same as butterfly coil. All right. So I was supposed to talk about electromagnetism, and it is based on these Maxwell's equations. that describe how the electric field E, or the E field, is produced by electric charge, rho, and also by the changing magnetic field B. I don't need to go into the details. And the magnetic field is produced by the electric current density, J, for example, current in the coil, or it could be also produced by the time derivative of the electric field. So that's the starting point, and I go to the basic principles. So in electromagnetism, we have the magnetic field and the electric field. The electric field, what this equation means, the first Maxwell's equation, is exactly this in the figure, that if there's a point charge, like a, say, electron, or in this case, proton, positive charge, the electric field would radiate in all directions in 3 dimensions. These arrows are the electric field. The electric field, what it means is that if you put another charge here, it will accelerate in the direction of the electric field. If this would be negative, it would be the other way, the electric field would be in the opposite direction. So that's the first Maxwell's equation. And for the magnetic field, if you have electric current, so electric charge flowing along this wire, then there would be magnetic field, it's red in this figure, that would go around the wire that carries the electric current. And if you have a ring, like in a round magnetic stimulator, if you have a ring of current, and the magnetic field, you sum up all the magnetic fields, it looks like this. So it goes through the ring and it goes back. Also, a refrigerator magnet, little magnet, produces this same shape of magnetic field. And then in TMS we have also electromagnetic induction. There's a changing magnetic field, and in this figure the magnetic field lines are the red ones, and if they change in time, like become stronger or weaker, there's the time derivative of the magnetic field, then there will be an electric field, according to one of the Maxwell's equations, that goes around. This is the equation here. This is Faraday's law, invented almost 200 years ago. So these were the Maxwell's equations. Now I will go step by step through the events that happen when this electromagnet is activated. So remember there are the turns of wire in this figure-8 coil in this case. These drawings here are from Jalinos, one of the authors of the first magnetic stimulation paper. This was written in 1997, A Guide to Magnetic Stimulation. And so this is a monophasic pulse, meaning that the current, in this case, goes only in one direction. So the current starts from 0, and the time scale here is milliseconds. So in about 100 microseconds, the electric current goes from 0 to maybe 3,000 amperes. Of course, this can be changed. And then it decays to back to 0 in about 1 millisecond. Be 3 kiloamps also. And this produces, the current produces a magnetic field. Of course, it has exactly the same wave shape. The magnetic field goes to something like 1 Tesla or 2 Tesla, depending on where you measure the magnetic field. So that's why it's called magnetic stimulation. We produce the magnetic field. But then there's the magnetic field, but the real effect comes from the electric field. So the electric field produced by Faraday's law, the formula is again there, is proportional to the time derivative of this magnetic field. And of course, the time derivative is maximum when you begin. So here is the time derivative. It's first in one direction, and then after you've reached the peak, the time derivative is negative, and the electric field will be in the opposite direction. The average electric field is 0 because you first go up and then you go down. So the magnetic field goes up and then down, so the average magnetic field is 0. And the average electric field is 0. And the amplitude, the strength of the electric field is typically 100 volts per meter in the brain. So here's again the induced electric field, and what the electric field does, it accelerates charges like ions in the brain, sodium, potassium, chloride, and so on. It produces electric current in the brain, and it is exactly proportional to the induced electric field. So here's the induced electric current in the brain. It's about 0.1 milliamps per square millimeter, depending how strong the pulse is and where you measure or calculate. So exactly the same waveform as the induced electric field. That's and the interesting thing, an important thing, is what happens when you have the electric current. It causes membrane depolarization. See, all the neurons have membranes, and when there's the electric current, it cannot go through the membrane because the membranes are electrical insulators. So the electric charge will be accumulated on one side, positive and negative on the other side, because the current moves away, the charge moves away from the other side. So there will be either polarization or depolarization of the membrane, meaning that the voltage is either reduced in depolarization or increased in hyperpolarization. Depolarization is more important here because it activates the neurons. And now, so imagine there's electric current that starts in one direction and then goes to the other direction. Then the membrane charge that is accumulated is bigger if you have the electric field for a longer time. So you start from 0 membrane charge change, and then you go to the maximum, and then when the electric field is the opposite, you come back. And after the pulse, after the 1 millisecond, there's no accumulated electric charge anymore. So this induced electric membrane charge is the integral of this original. This was the derivative of the magnetic field. Then you integrate, you get the original wave shape. So from the membrane point of view, it's really a monophasic pulse. So here I have this is the membrane, and when the electric current comes, these blue arrows here. There's an insulator. The charge cannot go through, so they are accumulated here close to they are practically, they're almost touching the membrane, but there's water and there's thermal motion. They're not exactly on the precisely on the membrane. They're a little bit apart from the membrane. And then on the other side, the electric field, which is also on this side, has caused positive charge to go away and negative charge come to the membrane, so the other side is negative, and therefore there's a change in the membrane potential. Then, what does the membrane depolarization do? It opens channels. Neurons have voltage-sensitive ion channels. Here's a depiction of sodium channel made of, it's made of proteins, and the lipid molecules here, and there's about 10 nanometers thickness, and here in the beginning it's closed, and there are charged molecules like the oxygen. When the membrane is depolarized, the electric field in the membrane is changed. Actually, it's very high electric field in the membrane, it's millions of volts per meter. and then this opens. The channel opens. Here it's closed, and here it's open. And what happens then is that the sodium ions, positive sodium ions, go through, and this causes action potentials in neurons. This is the waveform of an action potential as a function of time. And the action potentials propagate along, this is a picture of the neuron, propagate along the axon of a neuron. And when the action potential comes to a synapse, synapses are activated. And in the synapse, this is a simplified picture of a synapse. There will be post-synaptic, so-called post-synaptic potentials, but there's electric current going through the postsynaptic membrane, and this postsynaptic current produces the EEG signals that we measure. So in this picture, a pulse is given, electric current goes to the wire, the magnetic field is produced, electric field in the brain, the membrane is open finally, and then you measure with this EEG cap. And these signals that we show here are actually TMS evoked electric signals, EEG signals. Now, one slide about activation mechanism. So, as I said, that the membrane is depolarized in certain places. And there are several geometric possibilities, but the two important ones are that, let's look at this number one here. This is an axon which ends, so this could be an axon terminal. So if there's an induced electric field and current, and then there is a membrane here, then the depolarization happens here at the end. That's one possibility. Another possibility is that there's a bend in the axon before it reaches the synapse. So the electric field, these black arrows, go to the right, and so in a way one can say that the electric field current wants to go that way, but there are no membranes, so then there will be accumulation of positive charge inside here at the bend, and depolarization there, and the action potential can then start here. Actually going then, the action potential would then travel both to the right and to the left. All right. Yeah, please interrupt for questions if you want. Yes.
Just a question. So the major contribution to the simulation is the simulation of the axons. And what's the role or what's the contribution of the simulation of the cell bodies itself?
Well, actually, it is not exactly with certainty we do not know where the activation takes place, but there's good evidence that, computational evidence that the bands and also the nodes of Ranvier in the white matter would be places where the activation starts. Yes, the soma of a neuron, there's the dendritic tree, and if you have let's say that there's the cell body, and then there are the dendrites. So if the electric field comes from up, going down, electric current in the dendritic tree would come to the soma and make the soma more positive. And there's the axon hillock that is most sensitive to depolarization. So that is a possible and maybe even probable locus of activation also. Yeah. Then we were talking about the membrane. In the 1980s, the Nobel Prize winning research was done on the invention of the patch clamp technology and using it to study single ion channels. So there's a pipette, like a glass pipette, which is very so small, and you can, and this is the cell membrane, and you can grab the cell membrane so that there's only one. You may be able to grab there's only one channel here. And then you put electrodes on both sides, and you can change the voltage or current through this single channel. For example, sodium channel. The channel can open, then you can measure the current if it opens. And experiments have been made. And here's one example where the upper curve here shows the experimental manipulation. So first, the membrane was in the resting voltage, minus 65 millivolts, the interior being negative. And then, by putting a little current, you can change this voltage clamp. You can change the voltage to minus 40 millivolts. And there's time scale 5 milliseconds, and then you turn it off. And now, then with the patch clamp, there are 3 different repetitions of the measurement. You can measure the current through the single membrane. You see, the channels open not at the same immediately when the voltage is changed. Here it takes less than a millisecond. Here it takes more than a millisecond, and so on. So it's a random time from the membrane potential change to the opening of the channel. I believe it's because there's thermal motion. So the experiment is done very precisely. The voltage changes can be controlled very precisely, but there's random motion. So when you close the opening, then random motion of the molecules causes this delay to be different at different times. My former PhD student Larry Kopponen performed these experiments with electronics, which we have also here now. This is electronics with which you can control the magnetic field in a very flexible way. So here the magnetic field had this shape. The time scale is here, is 60 microseconds. So the magnetic field goes from 0 to the maximum in a certain time, maybe 70 microseconds, and then it stays the same for 60 microseconds, and then it is reduced. And here's the time derivative of this first curve. So first you have a steady change in the magnetic field, so the time derivative is about constant, and you have the maximum E field. And then the magnetic field is approximately constant, so then there is approximately 0 electric field, which means that the membrane polarization stays the same, and then there's the opposite phase. And what was manipulated here in this study was the length of this so-called hold period. How long was the, in this picture, the membrane potential was kept at this minus 40 milliseconds for a long time, like tens of milliseconds. But in our experiment, it was only either 60 microseconds, 30 microseconds, 15, or 2.5 microseconds. And the purpose was to study the channel time constant. So what is the question is, what is the average time it takes from the membrane depolarization to the opening of the channels? This is called the time constant. And it cannot be measured in the human, or not even in animals, in body temperature, because the very tiny and high resistance patch clamp pipettes, they have an upper frequency range. They're limited to frequencies below 100 kilohertz, and these are very fast events. So you cannot measure, so these measurements have been done at a much lower temperature, like the animals you can cool down to 15 centigrade or something like that. So now we are in 37 degrees human body temperature, and we can measure what happens in the human. And this is the results. So the motor cortex was stimulated, and motor evoked potentials were measured from the opposite hand muscle, and the vertical axis here, logarithmic scale, is the recording of the motor evoked potentials in the muscle. As a function of the electric field in the four different conditions. So if the hold period was long, of course, then you can wait for a longer time. So you need weaker electric fields. Just a moment. So when the hold period was 2.5 microseconds, you needed 125 volts per meter to get this intermediate MAP amplitude. When the hold period was longer, you needed less electric field. And from that, by plotting these four numbers here, we can determine the time constant, which happens to be 17 microseconds.
This data from a single subject, or?
There were several, I don't remember how many subjects, but this data is for one subject. I think, I'm not sure. I don't remember. This is already several years ago. Okay. Right. So we can, and this measurement cannot be done in with a patch clamp because it's so fast time scales. Not, at least not easily. Well, Victor said that I should be fast and to leave time for discussion. So I'm just saying that the focusing of TMS is not perfect. So this is the coloring here shows the electric field intensity when the target here is in the middle. And the one important thing that you cannot focus in depth. The strongest electric field in TMS is always, regardless of what, on the surface of the brain, not in the deep structures. Then one important thing, we'll hear more about this, and it has been worked here very much in this university here, the navigation technique. So you have cameras that follow the movement of the coil. The coil has these markers that shine in the picture of the camera, in the light of, and you can measure the location of the coil and orientation of the coil with about 1 millimeter accuracy. So this is, and then you can do this navigated TMS. It has been used in pre-surgical evaluation, so you can stimulate many locations in the brain and see where is the location that activates muscles. And then the neurosurgeon doesn't want to cut that part because then will be paralyzed. So TMS is intrinsically precise when we have this accurate navigation, but we also need to have a precise model of the coil. This can be done with a very high accuracy, so that you know the magnetic field very accurately, and then you need to know accurately with navigation of the head position and orientation of the head. You need a model for the head, conductivity model. You may need a cell model and activation model. I'll skip those. I don't go into the details now. Okay, how much time do I have, Victor? Plenty. Okay. TMS EG. Okay. So a long time ago, almost 30 years ago, actually in 96, we did the first combination of TMS and EG studies. And this was published in '97. So here's the brain, and we stimulated the left hemisphere at the cross. And the animation shows what is called the minimum norm estimate of the neuronal activity after the pulse. So it's a blurred, in reality it's much more complicated, electric currents, but this is a blurred image. But anyway, it shows that when you stimulate, this part of the left hemisphere is first activated, and 20 milliseconds later the right side is activated as well, because there are connections. Here's the real brain, picture of a real human brain. It shows the interhemispheric connections. There are about 20 million actions, neuronal fibers going from left to right and right to left, so it's no wonder that if you stimulate on the left, there will be an activation on the other side. It takes about, depends on what you look, 10 to 20 milliseconds before the activation is on the other side, or it can be also inhibition. And we can measure it. So this was nice because you could test how the stimulated location works, whether it's alive or not, whether it's, or too active, and you can measure the connectivity, whether the connections are okay. Well, you can also do it within. There are also connections within the hemisphere. Now, I'll explain a little bit about the brain signals. We published a book with the late Jukka Saarovas in 2019 called Brain Signals, both MEG, now I'm talking about EEG more. So what are they? What are the brain signals? Where do they come from? How to measure them? How to interpret them? How to influence them? These are spontaneous activity, alpha rhythm, and other signals. What are they? So one can say that they are action potentials and postsynaptic potentials, as I mentioned. Or you can say that they are electric and magnetic fields. There can be also other measurements of the activity. So these are the brain signals. The spontaneous activity at different, you can measure different situations. You can observe the alpha rhythm when you close the eyes, and you can measure theta rhythm, and delta rhythm at night, for example, and beta rhythm when you are, the brain is really active. So that's one, spontaneous activity. The other one is evoked responses, or event-related responses. And these can be measured when there are changes in the state of the brain due to stimuli or other events. We can give sounds, or visual stimuli, or tactile stimuli, or we can give a task to the subject, or whatever. So this is the point. State of the brain changes, then the brain, we can measure brain signals. What is a brain state? There are many definitions. There are many things that happen in the brain, state of the brain. The structure can change. That's one thing. When we learn, the synaptic strengths change. Maybe other reasons for their changes also. And then the metabolism may change, and hemodynamics. We are now interested in, when we talk about EEG, the electrical or activation state. One can also describe it as information processing state or excitability state. There are many other ways to describe the brain state. So the interesting thing in both magnetoencephalography, when you measure the magnetic field produced by brain activity, or EEG, when you measure the electric signals, is that the MEG and EEG measure a projection of the information processing state of the brain as a function of time. That's a statement, which I will explain. So here we need a little bit mathematics. JP is a symbol for the electric current in the brain at location R at time t. So this is the electrical state of the brain if you describe the internally generated electric current. It's called primary current in the brain. And the measurement, here I call it YI. There are many, I is an index, like 1, 2, 3. There are many sensors. I goes from 1 to, let's say, 64 as function time. It's an integral, it's a weighted integral. This is called the lead field. Here, Li for the sensor number I. It's a function of the location in the brain. So you multiply the primary current, the activation strength at each location with this sensitivity or lead field function. And what it, okay, what it means, I'm trying to illustrate in this figure, is that first of all, this JP, the primary current, has many components. Like you take one location, there's electric current in some direction, and other location, there's electric current. There's a list of current amplitudes. I cannot, it's a multidimensional current space. And here I have only 2 dimensions of the current space. And at each time, for example, time t1, there's a vector in this current space denoted by this arrow here. And because the brain state changes as a function of time, at the next instant, t2, it's a different JP. So there's a trajectory. I don't know this trajectory, but there is a trajectory. As a function of time, the brain state changes in the current space. And now, the interesting thing is that the measurement it can be described, the one sensor can be described by the lead field, which is another vector, it's the Li, it's the vector in this current space. This is the sensitivity vector. And now when the brain state changes, what we measure is the projection. So this dotted dashed line here, we measure, so at time t1 we measure the smaller amplitude, and time 2 a little bit stronger, and then we get the wave shape of the EEG. To me, it's amazing. We can measure directly the state of the brain. There's nothing to. It's like fMRI is indirect, but this is a direct measurement of brain activity. It's a projection. It's like the projection is the same as, you know, there's some object, and you see only the projection, not everything. You can then imagine what's the real object. So it's a little bit, it's exactly the same mathematics, but more dimensions in case of the brain. Instead of just 3 dimensions, projected on 2 dimensions. And okay, now this is just wanted to show one of these, my favorite pictures, and I think Victor and Dubé, and who will talk later, and Julio know this and sometimes make jokes about this, but this is how I think about it. So there's this brain state vector, which I just described, and it goes somewhere as a function of time. But what TMS does, it kicks the system to a new state. And then we can measure the EEG, what happens after that. Yeah.
So thinking about this, like it kicks the system into new state.
Yes.
Which state are you measuring now with the TMS? I mean, TMS is often regarded as the probe of the brain state.
Yeah.
So which state are you probing actually now? Are you probing the first one or the next one? Which are
The
you
next one.
measuring?
So if I do not give a TMS pulse, and I measure EEG, let's say 100 times, and take the average tends to be 0, because nothing, it's always a random thing happening if you do not stimulate. But if you give a stimulus, always the same stimulus, this kick is similar. And then you get a signal when you average this, do signal averaging. And then the EEG is not zero on the average because it tends to return to some kind of more probable state. We have one paper with Thomas Motanen, first author, where we were thinking about this, that the brain, it's also, you can think about statistical physics, the entropy. Like the brain, of course, is most of the time it's in these high probability states. It's very seldom in a state which is extremely improbable, right? So when we give a TMS pulse, it goes to a state that it wouldn't otherwise go. It's very improbable. So it has to come back to these more probable states. And this is what we measure. Get some information about the brain. It's hard to interpret the results, but this is what happens. We can discuss that. But it's, so how to measure the brain signals, with EEG or MEG or otherwise. And here's an EEG cap. This is the Bittium Neuro 1. And the EEG, you often see this kind of pictures. There's a head of the subject and the locations of the electrode. Electrodes, now, sometimes, very often actually in the literature, people think that they are measuring potentials, but they're measuring actually differences of the potential in one electrode compared to the so-called reference electrode. And the measurement that here Vj, it's a jth electrode, is an integral of the electric field. So you actually measure the electric field along, let's say, this red line, but the result is the same even if you measure some other integral, another line. So we actually measure electric field not potentials. If you understand this, think about it, you don't need to care about the reference electrode problem. There's no real problem, reference electrode problem. If you understand that it's the electric field that is being measured, and then equally important is the location of the reference electrode, real so-called active electrode and reference electrode. I can discuss that too, and there's a lot of discussion also in the literature and a lot of confusion. Then what I call fundamental equation of EEG and MEG is coming soon. So here's the picture of the brain, and there are these primary current elements. So there can be one pattern of current here, this arrow here, another pattern here, a third pattern here. They can be far away from each other, close, or whatever. And in this example, the first pattern has a time dependence, x1 times at time t, and x2, and finally the last one, x and s, where ns is the number of sources in the brain. These are called sources because they produce the magnetic or electric field. So we can describe the activity in this case in the brain with a vector x, which is simply the collection of all the activations in these ns sources. So that's a ns dimensional vector. And then we have the measurement vector, which consists of the Y1, Y2, for example, 64 electrodes, 64 signals, and what it is the projection. There's this so-called it's a matrix. It's the what's the name? Transfer matrix. Tell me. That's a standard name. Name for, pardon? Sorry? It's a lead field matrix also. Okay. It doesn't matter what the name is. It's an This is an unknown matrix, and X is also unknown, but we measure Y, and then there's noise also, N. Yeah, it's the mixing matrix. It's a mixing matrix. So it mixes, so if you measure from one of the channels, you get signals from all these sources at the same time. So the signals are mixed. The original activity the axes are mixed. So that's why it's called the mixing matrix. So that's the fundamental equation. Jukka Sarvas, that's a favorite. Julio remembers this. His favorite equation where you measure Y, and it's a set of numbers that you measure, and then you have the unknown X, but also this A is unknown. It's amazing that it can be solved in some cases. Okay, for example, okay, I'll go ahead. So where do the signals come from? They come from neuronal primary currents. Here is a depiction of a simple axon and an axon potential. When it moves from left to right in this picture, at a certain instant there's sodium. You know, the sodium channels have opened and sodium flows in from both sides, all sides. So the interior will become more positive here because positive ions go in. And when this is positive, then there's electric field to both left and right, equally large to the left and right. So the neighboring area will also become positive because the current flows there, and the channels will open here. So in a while, this part will be activated and there's propagation. Now you see that the electric current pattern here is symmetric. There's equal current to the primary current to the left and right, so this doesn't produce a strong electromagnetic field. So we do not measure this with EEG or MEG from the outside. Now, however, we measure the synaptic current. So this is the synapse, and when the action potential signal comes, vesicles release their transmitter molecules, like glutamate, and that causes the receptor molecules to open the channels, and there will be electric current from through the membrane and inside, and then they go through the membrane, so there's this length constant. Current doesn't go to infinity, it goes out. The length constant describes how far they go. Then I have this old picture of mine, so where I describe what the primary current really means. So here's a little more complicated synaptic geometry. There's the axon and postsynaptic currents. So the primary current is not to the left, although the synaptic current is to the left, because the postsynaptic neuron in this case does not allow the current to go to the right because there's the membrane. The current would go down, so therefore the primary current have this kind of shape instead of just the transmembrane current. Just a little detail here. Then how should we interpret these EEG signals? We have to solve the inverse problem. So the forward problem is that if we would know the signals in the brain or the primary currents in the brain, the neuronal currents in the brain, we could calculate the EEG signals. That's an easy problem if you know the geometry and then electromagnetism. Solving the inverse problem is it's not an easy problem because there are mathematically many allowed solutions. But anyway, you can model neuronal activity, you can estimate the parameters of the model, you can correlate with other information, you can model at a higher conceptual level, and you can estimate higher level parameters. I'm just mentioning these. And then this slide I want to show, because sometimes people want to know, for example, where the activity takes place at a certain time after a TMS pulse. And people often want to have an estimation method that is the best possible. So there's books on estimation theory, and this is the logic. There's experimental data. And there's also a priori information. So you know something about, like, you, for example, you know that the activity is in the brain. It's not somewhere else. But there may be other kinds of a priori information. And then there are the laws of physics, in particular electromagnetism, and experimental parameters. You know when you gave the stimulus, for example, and where. So there's a priori information, and then when you combine this information from above, you get a a posteriori probability distribution. This is the Bayesian logic. In principle, this can be done. It means that what is the probability of different answers? And then you need a definition of best. What do you mean by best? And one definition is that the expected error, because there's noise and many uncertainties, the expected error should be as small as possible. Many people think that that's a good definition of best. And if you can define mathematically what's the definition of best, you can get the best estimate. That's the minimum norm estimate. Is one best estimate with suitable definitions, and then there's no way to make better than the best estimate. But the best estimate depends on the definition of best and depends on a priori information. And so the result can be different depending on what a priori information you have. All right, and 5 minutes, did you say? Oh. Okay, less. Okay, well, no, oh, okay, good. How can you influence the brain signals? TMS, that's what we're interested here. And a good example is this, also one example. This was the Massimini et al. Science paper where they stimulated the brain close to the midline, I think frontal cortex, and so during wakefulness, there was a complicated response, EEG response, and during deep sleep, non-REM sleep, a strong response, but very simple. And I'll just show the video. During awake, so stimulus was given here, and the electrical activity is moving around to the frontal cord, to the other side of the brain, the front and to the back, and it's very complicated and takes a long time before the signal dies away or becomes more random. And then if you look at exactly the same stimulus when the same subject was asleep, there's a very strong response in the region that was stimulated, but it doesn't go very far. And the finding was really that during deep sleep, the different areas of the brain are not well connected. Signals don't travel so effectively. Well, there are artifacts in the EEG signals. I'll not go into details, but I'll just point out that Tuomas Mutanen, after the work of Hanna Mäki, developed the sound algorithm. So on the left, you see beautiful EEG signals. They look beautiful, but there are some artifacts, and with this sound methodology, you can clean it out and get beautiful signals from whole channels. So many people have contributed to this work, and I'm afraid I have forgotten something, but it's a big effort. This is in the Connect to Brain project, and in addition, we have a collaboration, for example, with Sao Paulo University. So thank you very much. Okay.
have one. So you did show that we are not stimulating directly the neurons because we are just accumulating charge in one side of the membrane and in the other side. So we somehow push the ions that are already in the exoplasm or in the body of the cell to somehow create a local electric field that will depolarize it. And they just made a very back-of-envelope calculation. So if you get like 100 volts per meter, and if you multiply by the thickness of the membrane, that's about 10 nanometers. So you get about 1 microvolt. And 1 microvolt, we know, is not enough to depolarize the cell because we need some millivolts. My question is, can you visualize or envision a way where we can produce a magnetic field that's 1,000 times higher? Because then we'll be in the range of millivolts, and then we probably will open the channels directly. And this would be a very different change in the game, I guess. I would like to have your comments.
Yeah, the direct electric field, 100 volts per meter, has practically no effect directly on the ion channels. It's some, but you know, as a function during time, the charge will be accumulated, and then the internal electric field changes on the order of 1 million volts per meter. And you're asking whether we could have a very brief pulse with extremely strong electric field, like 10,000 times stronger than now. I don't know how that would be possible technically. Except by one could shine light, which is also electric field and magnetic field, directly to the molecules and cause a transition, you know, in the energy levels of the molecules. Of course, that would be possible. And something like that is actually done in optogenetics. Yeah. And in the eye, we get electromagnetic radiation, and there's a very brief, it's at the order of femtoseconds in the eye. There's a strong electric field. You may want to calculate that. That changes the receptor molecules, what are these, rhodopsin molecules. And something happens, and that can also done in optogenetics. Yeah, actually, it has been solved.
Thank you, Professor Ristov, by your talk. Actually, is a curiosity in the other direction that Professor Baffa notes here. My question is related not in terms to increase the magnetic field, but in terms to reduce the magnetic field. Because I saw in the, I don't know, in the last 2 years maybe, some equipment that say they are portable, this kind of, yes, it's a portable TMS. And they are saying that they don't need a very high magnetic field in terms to induce an effect in the brain. And I would like to know your opinion about that.
Well, if you have, it depends on how weak they are, but there are some instruments that have been published in the literature where the stimuli are so weak that the channels do not open. So I don't know what that's useful for. On the other hand, there are some intermediate levels of the magnetic field, like 10 or 50 times smaller than what we now have. So they may have an effect in sort of a random way. You see, when we think, when our brain is active, the channels open, you know, and some of the channels, in some places of the neuron, or in some neurons, the membrane potential is already before the pulse, close to the threshold. So then you need a little kick to bring it to the threshold. So maybe that will be useful. It's a really interesting thing to study. But if these are extremely small, then even that wouldn't work, I think. That's my opinion. It's only an opinion.
Thanks for the good talk. TMS is used for therapeutic function. So in that slide that you showed about the brain state, in that case, the subject's state is a pathological state because you were applying. So when you apply the stimulus, this TMS stimulus, you change the brain state to a different one. And of course, you don't want it to come back to the pathological state. So the ideal thing would be that the TMS stimulation is somehow it reconfigures the entire system so that, of course, it goes to a different state and then it tries to come back to a default state, a stable one, but not the pathological one. What's your opinion about that? Is it possible? Do you think that really it reconfigures the entire brain state?
Yeah, this is really important question. So when I didn't talk about that there's plasticity in the brain, meaning that the synaptic strength can change. For example, it is well documented that if you have two pulses coming to the same neuron, there's this spike timing dependent plasticity, and the synaptic strength change. So that after the pulse, although the activity has come to the normal, the structure of the brain in terms of synaptic strength will change. And the so-called energy landscape, this is related, I think, well, I'm sure, to the Hopfield net thinking, where there's this energy landscape and there may be these harmful attractor states, and you want to get rid of those by stimulating in the right way, by changing the topology or connectiveness of these neurons. But how it should be done is not well known. Well, people trial and error at the moment. People have tried different things, and some of them work, and we want to understand the brain better so that we could do better therapy. This would require much longer discussion, but I don't know if this was okay.
So you talked about the energy landscape. How this energy landscape is stable across individuals and between with the same individual between like different conditions?
Well, that's a really thing. It brings to a very interesting field of study the criticality of the brain. We are all the time at the brink of collapse, and there's this homeostatic set of mechanisms that keep us from collapsing in the let's say mental states or whatever. Because we want to be both stable and able to adjust to environmental changes. It's really important to learn new things, so that we're not the same tomorrow after this lecture before. Yeah.
Thank you very much for the nice lecture. I have a quick question about the opening of the channel. You showed that when you change the potential, the channel opens and it closes after very quickly.
Yeah.
My question is actually two quick questions. The first one, what is exactly the condition for the channel to open, and why it closes after a short time? Why it does not remain open?
Well, the Huxley-Hodgkin equations, it's also won the Nobel Prize, describes the kinetics of the channel opening and closing of both sodium and potassium channels. The mechanisms are like physical molecular mechanism, and when you have a strong electric field, there simply forces on the atoms, which are not like the I haven't studied this, but for example, oxygen atoms can have a negative charge in a molecule, protein. And then when you turn off the electric field, the molecules tend to fall into a lower energy state. That's the answer, I think.
Yeah, Rista, you talked about the fundamentals of the TMS, and so you didn't touch too much the stimulation modalities. You know, there's paired pulse, RTMS, ITBS. So and we saw over past couple of years, a decade actually, the development of these stimulation modalities and very different effects to the brain states and to also these pathological brain states. Do you think that in the future we will have some new TMS modalities with new possibilities, open up new possibilities of treatment research and so on?
Being in Brazil, I thought about immediately after your question of football. So somebody kicks the ball, doesn't go to the goal. You need another kick. Somebody makes the goal. So it's the same thing. And you have to study it. The situation is always different. It's not always, it depends on everybody moving back and forth. So I don't know really what your question was, but this is an important thing, that you first kick into the brain, brings it to one state from where you can kick it to the other.
Yeah, but okay, we can argue this, and you're always right. Yes, the way you kick goes to the goal, So
the last one, but the first is to prepare to the person who makes the goal. So, or the TMS pulse that makes it to the right place. Yeah. The two pulse stimulus they are powerful because you prepare the brain for the next pulse, right?
No. So I was kind of mixing these two previous questions into you have the lasting effects of the TMS. So how long does it last? But then you also have the modality of how you apply the TMS pulses, and then you have different lasting effects. The easiest one is the RTMS with the low or high frequency, and then you have inhibitor excitatory effects. But then you have ITBS, which is coupling to some other brain dynamics, to oscillations, for example. And then you have the multilocus TMS, So I work with multilocus TMS, but I don't know yet what to expect from it, And do you?
Yeah, my knowledge is very limited, but I think we are in you are now in a very important area, and we should study the papers and we should try to understand, because this is really the future, to learn what sequences we should give. If you give one pulse, the main effect locally is inhibition, because we stimulate both inhibitory and excitatory neurons. And the inhibitory neurons stop the activation of the excitatory neurons. And this is experimentally found. There's the cortical silent period if you just give one pulse. But after the inhibitory first pulse, well, there's excitatory also, but the effect will not stay inhibitory if you immediately give another pulse after 20 pulses per second, for example. So you can keep the activation up even if there's inhibition, if you just keep activating all the time. So that's one fundamental difference between single pulse and rapid rate stimulation. But I think there are better experts here to understand that. But and we should learn as a community to do things better now. There are huge possibilities.
Thanks, Professor Risto, for the talk. You showed us an animation where you apply the pulse and you see the EEG produced by the TMS pulse, okay? So I'm interested in knowing how this trajectory changes from one subject to another. I think that is not very clear for me, because I was thinking about the question of Professor Hock, the idea of bringing the brain activity to a healthy state. This makes me think that the trajectory that you see when you apply the pulse also tells you about the brain state in which the subject is now. So it kind of gives us a way of identifying the brain state.
Right, yeah. In the individual, it tells you about the state of the brain, the differences between the subjects, and for example, Marcello Massimini and his group, they have studied the EEG responses in different states of consciousness, for example, anesthesia or coma or vegetative state, and you can even measure what the conscious state seems to be. Yeah, so the responses are different in different people, but there are many similarities, of course. For example, if you stimulate the left hemisphere, most of us have similar connections to the other hemisphere. So they're both similarities and differences, especially between health and disease, but also between healthy people. And there are many studies of, for example, language, that people are really different in how we think and then formulate our thoughts as language. So some people think of verb in words, and some people think without words. So they have more difficulty in converting the thoughts into language. They're equally smart, but it takes time to express it in language. So, yeah.
Thank you for this.
Any other question? So I have two important announcements. First, today is the first day of spring, and we have rain. So finally, and because we have rain, the coffee break, you'll be served here inside. So we have just to go through this door. So we already have coffee break. Okay. Thank you, and that's adjourned for half an hour, and then we resume.
Thank you.
bensound.com. 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 bensound.com.
Okay, welcome back. It's my pleasure to introduce Professor Antonio Roque. He's an associate professor in the Department of Physics in our faculty here in Ribeirão Preto, where he founded and coordinates the Neurosystem Laboratory. His research focuses on computational neuroscience with an emphasis on biologically plausible models of neurons, astrocytes, and their interactive networks. He has also worked on animal behavior modeling, complex networks, and medical decision support systems. He's active in training of researchers, advising undergraduate, master's, doctoral, and postdoctoral students in the physics applied to medicine and biology, and computing programs at the University of São Paulo. Today, he's presenting the talk From Neurons to Function, How to Connect Basic Neurosimulations to Brain Functions. Thank you very much, Professor.
Hello, and BAF and all the organizers for inviting me to this talk. I must say at the beginning that I don't work with, is it okay? Yeah, close it, okay, yeah. I don't work with TMS, but I'll try to entertain you. And there'll be some TMS at the end. So that's the title that, well, actually BAF suggested the title to me. So let's see. Well, one of the main challenges for modeling the brain is that the brain is a multi-scale system. Oh, sorry, I don't have a pointer with me, so I will be probably pointing with my finger or the mouse pad. Yeah, you can. Oops. Yeah. Actually, I don't have one here that I can. Well, let's see how it goes. So the brain is a multi-scale system. It covers all of these spatial scales from, say, channels at molecular level to the whole brain and the whole individual. And this makes it complicated to model the brain and understand this highly sophisticated system that makes us as we are. But the idea of computational models is that they help us to understand the complex behaviors that emerge. Thank you. Yeah. I think that's better to use this. Yeah. They help us to understand the complex behaviors that emerge out of these complex systems. One of the things that I always say about models that is not so obvious is that models are repositories of knowledge that we gather about the brain or any system which is simulated in silico. So you get the data from different experimental modalities. It's difficult to bridge them because they are obtained with different techniques. But if you put them into a model that has a structured language or code that uses that knowledge to simulate a system, you actually are storing them in that model. So it's a way of keeping them and testing how they interact with each other to provide the behavior that you're supposed to understand. And by running in silico experiments or experiments in the computer, you can test several hypotheses that are difficult to be, or impossible, because in some cases you cannot intervene in a human being to be verified experimentally. So that's the promise of modeling. And models, of course, they are deliberate simplifications of phenomena. If you want to construct a model, you deliberately simplify it because otherwise you'd be using the entire system. That's the paradox of the map and the territory. So you have to simplify it in order to make it useful. But they provide an intuitive understanding of the processes modeled. They are helpful in relating hypotheses to experimental data. They integrate data from different levels. That's one of the good aspects of models that has to do with that spatial scales that I mentioned at the beginning. And of course, the most important thing is that they can predict new phenomena that can be tested experimentally. So that's as I said, the promise of the models, a good point about models. But of course, when you want to model something, you have to face this difficult task of choosing the level of modeling. There are basically two modeling levels there. One is so-called detailed. Some people prefer to say realistic. I don't like this word realistic. I prefer detailed.
So detailed models have many variables and parameters. Simplified models have few variables and parameters. And both of them are useful to understand the system. And the choice of modeling level depends on the problem and also on the skills of the researcher. There are researchers who are more mathematically inclined, so they prefer simplified models. The ones who like more, say, who like computers, they prefer to use more detailed ones. And also depends on the available computational resources. Of course, if you are a clever researcher and you are in a place where you don't have powerful computers, you have to work on simplified models. You have to choose, say, the right type of modeling for the environment where you are. So I have this kind of three-dimensional representation of a network model in neuroscience. So any network model in neuroscience is composed of three elements. A neuromodel, so you have to choose, and I'm gonna, in the next slides, I'll show you several different neuromodels, single neuromodels that can be used. So you have to choose the neuromodel. Of course, neurons are connected via synapses, so you have to choose the synaptic model you're gonna use in your network model, and then you have to choose the network architecture, how you're gonna put those neurons and synapses together. And this creates a problem because, as I say here, there are almost as many network models as researchers in the area. And this makes it difficult for having communication between them. Researchers usually are focused on their own model. They don't like to hear about other person's model. So in a sense, there are few unified network models, and this is one of the challenges for the progress of computational neuroscience. Computational neuroscience, or say brain theory, is in a stage, compared say to physics, is in a primitive stage compared to physics. We are kind of in a pre-Newtonian stage, if you want, compared to physics. Think about the, say, the early pre-Socratic philosophers. Each one had their own understanding or model about the brain, and it was difficult to unify it. And only after centuries, so those giants of physics like Galileo, Kepler, and Newton, and so on, provided unified models that helped us to understand the world around us. Copernicus as well. Forgot to mention that. So let's go to a very short review of neuron synaptic and network models. There'll be some equations in those slides. I ask you to please not pay attention to the equations. Otherwise, you're going to not pay attention, not focus on the content. So to start with, there are two basic different types of neuromodels that one can choose. So-called spiking models, which are neuromodels that have as their output spikes as real neurons, and also firing rate models, which have as output not spikes, but a function that captures the firing rate or the short time average activity of a given cell. So you imagine that you have a neuron that has having lots of spikes as output, and then you average these spikes over a short time interval. And then you have some, if the neuron is really active, you have lots of spikes. If it's not so active, it has low spikes. So you have an activation function that captures the relation between the firing rate and time. So those are basically the two types of neuromodels that one can have. So when you hear a talk or a paper where they say, I have a spiking neuromodel, that's what they mean. They mean that spiking neuro, so a model that has spikes, where the cells, the units in the model have spikes. The first neuromodel that at least that's known in the literature is the one proposed by Lapic in 1907. It's not so well known, but it was the first integrator-fire model. Of course, it didn't have at the time, it was basically a circuit model composed of a capacitor and a resistor in parallel. And then when the capacitor was charged due to input that it received, and when the voltage reached the threshold, the cell would emit a spike. And the spike, of course, would be put by hand. Those are figures from the original paper. has been translated to English, if you try and find. The model was mostly forgotten for about 50 years, and then in the 60s, Knight in England, in UK, rediscovered it and christened it as integrated fire, because that's what it does. And it became very popular from the 1990s onwards. Today, I think it's the most popular simple neural model used in simulations. Some years later, in the 1940s, there was this very influential model by McCulloch and Pitts. It's the first model that showed that if you couple neurons via connections that are weighted by some parameters that were called synaptic weights, and the neurons are binary cells, so they can also provide, say, 0 or 1 output, depending on the input they receive. They showed, by proving a theorem, that any finite logical expression can be implemented by this model. I mean, they were the first one to show that networks of neurons can compute useful things. And they originated the field of artificial neural networks, or artificial intelligence. They were not so very influential in the field of computational neuroscience, but in the area of computational science, they were really fundamental, foundational. And of course, in the 50s, accomplishing a work that has been conducted by Hodgkin and Huxley since the pre-war years. Hodgkin and Huxley in the UK, they proposed a famous Nobel Prize winning model that explained how an action potential can be generated in a cell membrane due to the opening and closing of the conductances, ionic conductances present in the membrane. And in that case, in their model, since they worked on the giant axon membrane, they considered sodium, potassium, dynamic conductances, and also they had a leakage conductance that represented the effect of all the channels there. It's a 4-variable model, which for mathematicians is already complicated, so it's a detailed one. It's considered a detailed model. Difficult to understand because of its non-linearities. But it originated a procedure to simulate neurons, which is called conductance-based approach to neuromodeling, because the model is based on conductances, on dynamic conductances. So any model that adopts the Hodgkin-Huxley formalism is called a conductance-based model. So basically, it's conductance-based because the ionic current that stimulates the neuron has conductances there that change in time, according to some equations. You don't need to pay attention to them. And luckily for us, this formalism was able to capture the behavior of most neurons, not only the giant squid axon, but also mammalian neurons, which are really important for us. Because they can be described in the same way for most cases. And this, knowing due to the advances at the time of Hodgkin-Huxley, they didn't know about ionic channels. Only in the 60s they were discovered and studied, and their kinetics were determined, and people discovered that there are a large range of ionic channels, the zoo of ionic channels, but all of them can be modeled via the Hodgkin-Huxley formalism, and they can be understood. The functional aspect can be determined and simulated in computer. And later in the 60s, Wilfred Rowe in the US proposed an approach that allowed the modeling of neurons with spatial extent, because Hodgkin-Huxley model is a point neuron, has no spatial extent. But if you consider compartments where the ionic channels are distributed in these compartments and they are connected via resistances simulating the axial resistance of a real cell, you can put them together like a Lego play, and then you can construct neurons with sophisticated morphologies, and you can study the propagation of potentials across those, the dendritic tree of a given neuron. This is a Purkinje cell model, one of the first one that has been studied using this approach, and then you can say understand the nonlinear interaction between synaptic postsynaptic potentials across the dendritic tree of a neuron, and how they eventually sum up and produce an action potential at the soma or axon hillock, or even dendritic potentials as well in case of active neurons. So they are really they're very high-dimensional models, because some models like that have hundreds of compartments. Each compartment has at least 4 equations, but in general they have more, because they have more ionic channels. Remember that the zoo of ionic channels I've shown to you. So they're really complex, and only via simulations they can be understood. But then, to simplify things, those Fitzhugh, in the 1960s, he noticed that the Hodgkin-Huxley model can be reduced to a 2-dimensional model. This is because out of the 4 variables that are, out of for the 4 variables that are present in the Hodgkin-Huxley model, the voltage variable and the activation variable for sodium, inactivation for sodium and activation for potassium, 2 of them are fast and 2 of them are slow. So you can reduce them to a model with only 2 variables, a slow and a fast one, which can be represented in a phase plane. And then, since it's now a 2-dimensional variable, you can use dynamical systems tools to determine the fixed points of the system in this 2-dimensional plane. The points where the system is stable, and study perturbations around those equilibrium points to determine the stability, and then study the excitability of the model. And this was really important, because it allowed through the works of many other researchers from the 60s until the early 2000s, the understanding of the different mechanisms that give rise to not only a single spike or action potential, but to trains of spikes. So there are different mechanisms. All of them have been, say, mapped using those dynamical systems tools for this, not only for the Fitzhugh-Nagumo model, but for other models that are also simplified, following the paths that were inaugurated by Fitzhugh. And Nagumo, by the way, was an engineer. He was the one who implemented the Fitzhugh-Nagumo in hardware. So that's why today the model is called Fitzhugh-Nagumo. Another influential model that opened a different pathway or avenue in neuromodeling is the Wilson-Cohen model. It's a rate-based model, so it's not a spiking model. So the output of neurons are continuous functions of the inputs, and it's a population model. So each cell in the model, it doesn't represent a given neuron, but represents a population of neurons. In the first version of the model, they had all of them lumped into, two populations, excitatory and inhibitory, interconnected. The second version of the model is an inhomogeneous version, so it had variations in the behavior of those cells across space, giving rise to the concept of neurofield. And of course, the main researcher behind the neurofield modeling is Amari in Japan, who produced similar models to Wilson-Cohen in the 1970s and 77. And the model was interesting because in the early stages, it was studied by then, and it showed that they can produce a large diversity of dynamical behaviors. Today, this model is really popular because of fMRIs, which are, so as you know, techniques that allow us to visualize the brain at a coarse, relatively coarse spatial scale. So the population models are more adapted to this type of data than neurons, than models that consider individual cells. But then coming back to the simplified models, the 2-dimensional models, then there were proposals of nonlinear integrating fire models, which are basically the same integrating fire model that one had before, the one that was proposed by Lapic, simply an RC circuit, where the The function of voltage that appears on the right-hand side here is not linear, is a nonlinear function. So this gives rise to a nonlinear integrated amplifier model, which is able to produce different types of spiking behavior. So it's a richer model. And following the path, Izikevich, a Russian researcher who is based in the US, and Breithoroughs and Gerstner in Europe, they propose more or less at the same time 2 different models that generalize those nonlinear integrated amplifier models by adding a second variable. So the model became a B-dimensional, a 2-dimensional model, and showed that by considering those 2 dimensions with specific nonlinearities. In the case of Izikevich, is quadratic. In the case of Breakthoroughs and Gerstner, which the model is called ADEX, is exponential. There's an exponential term here. ADEX is adaptive exponential element. They show that by changing the parameters of the model, one is able to reproduce basically all kinds of spiking behavior of non-neurons, or at least non-cortical neurons. So since their work, this work opened the way to simulate networks of neurons that have different spiking repertoires in a relatively simple way in terms of computational power. You don't need to simulate those large compartmental models that, like the one I showed to you, to have spiking behaviors that are similar to the ones observed in real cells. And Izikevich showed this in this famous diagram showing that the implementation cost of simulating their model, and I put ADEX here because it wasn't known at the time. You can have not, of course, a high degree. Of course, you have a high degree of biological plausibility in terms of mimicking the behavior at a low computational cost. So this is opened the way to simulation large-scale networks of those neurons. So to summarize at this point, neuromodels, detailed versus simplified ones. You have the conductance-based models, which are Hodgkin-Huxley-like, with compartments. They are useful to study single neurons. So they are really useful for this to understand the function, since the title of the talk is about function. So those models are important for understand the function of single neurons. So they allow to the understanding of the propagation of the membrane potentials along the dendritic tree. They are useful for studying pharmacological manipulations, all in silico investigations. But to model neural networks, they require knowledge of all the parameters for all the neurons present, which is a phenomenal task. It's really difficult. So in general, what people do is to estimate parameters. So they deviate from the promise of having a really detailed model. On the other hand, the integrated amplifier models and its variants, they're too simple to be used to understand single neural models, but they're really useful for understanding for simulating large-scale networks. And say, understanding the functional properties of those networks. Then coming to synaptic models, basically the idea of a synaptic model is to give in a presynaptic spike to simulate the postsynaptic potential, which could be either excitatory or inhibitory. And by doing this, one would add synaptic current to the equation that governs the voltage of the cell, of the postsynaptic cell, and the current would simulate the behavior of the presynaptic cell. And the main idea would be to change the synaptic conductance in a way that mimics the behavior of the postsynaptic potential. In general, people use fixed functions to simulate the conductances. For integrated amplifier models, there are basically 2 types of synaptic current models. One is of synaptic models. One is so-called current-based model, in which the synaptic current is simply given by a fixed parameter, the weight of the synapse, multiplied by a fixed function. So this is a current. In the other type, which is called conductance-based model, one has also a weight, but the dynamics is in the conductance. This is more realistic in a sense because it depends on the difference between the current voltage and the reversal potential of the cell. I can talk about the details of this later if you want. Then one can simulate short-term synaptic plasticity. Short-term synaptic plasticity has to do with the fact that when the presynaptic cell is active, it's using neurotransmitters, so it loses resources that can be used to activate the postsynaptic cell. So this is depression. But at the same time, since it's active, calcium enters the membrane of the presynaptic cell and gets accumulated there, and this facilitates the occurrence of new of the release of neurotransmitters, of quantum of neurotransmitters by the presynaptic cell, and this is facilitation. The most popular model for this is the Sodex's macro model. It's, the equations is there, but I'm only mentioning here because I'm going to mention this work later. Then if you want to model long-term plasticity that has to do with memory that lasts for minutes, hours, or the entire lifetime, the main paradigm is the Hebbian plasticity, neurons that fire together, wire together. They have been experimentally verified, and this gave rise to the spike time independent plasticity, STDP. And the way to model that is via changes in the conductance that lasts for much longer time than the dynamics of the voltages and the other conductances that I mentioned before. And coming out to the types of network architecture, there are basically three types that the modeler can choose: artificial, biologically inspired, and data-driven. Of course, this is my classification. So an artificial architecture is that one that tries to capture the generic pattern of a given circuit or network, that brain network that the modeler wants to simulate. Could be a random network, could be a small world, scale-free. So people who work on network theory know all of these, hierarchical modular. So you have lots of dimensions there, out of which you can take, you can choose the architecture you want. And then you organize your network according to this scheme. They can provide useful insights, but of course they are made out of artificial architectures. They are biologically inspired ones because they incorporate elements of real network architectures, not in the very detailed ones, but so they have some elements. So neurons with dendrites that have connections that resemble the real thing. This is a model of primary somatosensory cortex. This is a model of the olfactory system. This is a model of the primary motor system. They're all of the type they call biologically plausible, because they are not really too detailed, but they capture elements of the architecture of the real system. And they're data-driven networks, which incorporate quantitative, that's the main difference here, quantitative information on neurons and our connections. And they could be also, they could be divided into models that address local brain circuits, the microcircuits, as people call. So they focus on the microscopic scale. There are some examples there. I would just mention here the model, the deep brain modeling approach that has just finished. It was funded by European Union in the past say 15 years. Resulted in a very detailed model of a cortical column based on data gathered from different animals and used lots of different techniques. That was the Blue Brain project. So, that resulted in this model. Very difficult to be understood, but successfully implemented in the computers available. Then one can use quantitative data to construct macroscopic scale models, which cover different brain areas. This is a model of the visual areas of the brain of the macaque monkey related to vision. So each module here is a microcircuit model, which by itself is a data-driven model. developed by Porchens and Diesmann in Jülich in Germany, and then they connected those modules into this network that covers most of the brain, and they can simulate it and try to understand the behavior. So let's kind of summarize everything that I said up to now. This is the first part of the talk. At the moment, the past, we are just changing eras in neuroscience, if you want. We have most of the efforts by theoreticians have been dedicated to single neural models, all those models that I reviewed very, very briefly now, and they have addressed functional questions related to single neurons. Single neurons have been really well scrutinized and understood via those techniques, both detailed models, which like Hodgkin-Huxley model allow the understanding of the role of ionic currents and dendritic processing in the behavior of single cells, and simplified ones allowed us to understand. Only 10 minutes? Yeah, okay, for the questions. Okay, 15, okay, yeah. And the simplified ones allowed us to understand neuroexcitability, the mechanisms behind neurospiking, while the network models focus on anatomical fidelity and computational implementation, but not so much on function. So the real challenge for modelers was to implement the model using those quantitative data and optimize the computers and code used to that. And you don't find many functional questions out of those, say, the Blue Brain project or the other model that I mentioned to you. But in the future, since now neuroscience is moving through a kind of real paradigm shift in the area from, say, single unit measurements to multi-electrode and optical recordings and all those imaging techniques that allow us to probe population level questions, one can use those models, those large-scale models, to address those questions, especially because we have now those simulators, NeuroMat, Nest, which have been around for, say, 20, 30 years, but now they are really got to a position where they are user-friendly and more available to any kind of modeler, not only to, say, engineers or physicists, but even to biologists. So they can be used as modeling tools for them to address functional questions. And they save time from researchers because you don't need to write a model from scratch. And I know that physicists like to do everything based on their own knowledge from scratch, but it's important to get together with other researchers in the area and collaborate to the construction of those open source tools. So now for the, say, 10 minutes that I have left, I'm going to give an example which is based on the, as in Brazil we call, scientific initiation project of Rafael Gigante, who now is starting his master's with me. He's here. He'll have a poster on Wednesday, so for further details, please ask him. But his model, his project, which was developed over 1 year, is to re-implement this model here, which is a cortical model for the primary motor cortex to study deep brain stimulation, the effect of deep brain stimulation in a thalamocortical microcircuit model. The original model, which is from 2021, sorry, was implemented in MATLAB. Rafael re-implemented it in Julia, which he's more, say, acquainted with, and perfected some of the aspects that the original model, which were obscure in the original model. So it's a good re-science paper, if you know that journal. So deep brain stimulation, of course, it's not TMS, but it's the closer I can get to TMS in Parkinson's disease. So Parkinson's disease involves excessive beta band oscillations in the motor cortex and also in the basal ganglia, and these pathological links, rhythms are linked to motor impairments, all the abnormal behavior that one observes in Parkinsonian patients. And high-frequency DBS is applied to this subthalamic nucleus, which is a basal ganglia structure, to alleviate those symptoms. It reduces the excessive power in the beta band but the mechanisms of action are still unclear. And evidence suggests that DBS also affects cortical structures, not only the basal ganglia structure where it is applied, via this antidromic activation. Antidromic is because it propagates in the opposite direction of the usual signal that comes from soma to axonic terminal. So this is the thalamocortical basal ganglia loop, and this is the antidromic direction. So DBS is applied to the subthalamic nucleus in the basal ganglia. This is the basal ganglia. There are two main pathways that connect the cortex to the basal ganglia: the direct pathway here in red, the indirect pathway here in blue, and the hyperdirect pathway is the one that goes from cortex directly to the subthalamic nucleus and other structures in the basal ganglia and back to the cortex via the thalamus. So the evidence that I mentioned to you, that some papers that show that, DBS can also produce activation of the cortex via this propagation of activity in the opposite direction of the usual soma to terminal, the soma to terminal direction. This is called antidromic in opposition to orthodromic. So the hypothesis for this to happen is that because of the interlaminal connections within the cortex, there could be generation of activity in the cortex and modulation of this DBS-induced activity, and this would lead to the reduction of the beta power. And also the thalamus would be involved because of the reciprocal connections between the cortex and the thalamus. So the thalamic cortical loop is considered to be an important structure for this modulation. So those guys there that I mentioned to you, Farokini and Lowery, they constructed this model to address this. But they actually implemented a model that is based on this sequence of works by the group of Carl Friston in the UK. So they basically consider different types of thalamocortical loop models, as you can see here, where, say, you have no, they started to consider just a biologically plausible model of the M1. The structure of the primary motor cortex, as any cortical area, has 6 layers, including a granular layer, the layer 4. But in that case, they lumped all of the cortical neurons into 3 basic layers: superficial, middle, and deep. All of them composed of pyramidal cells, which are excitatory, and a 4th cortical layer composed of inhibitory cells, which they call cortical interneurons, which provide common input to all 3 excitatory cortical cells. And also they had a thalamic modulus model coupled to the cortex, which is composed of 2 structures or 2 populations. One of thalamic relay neurons, which are excitatory cells that relay information from thalamus to cortex, and thalamic reticular neurons, which are inhibitory interneurons. But how those structures are connected, and what's the effect of those connections into the reduction of beta power? That's the question they wanted to address. And by using Bayesian inference, a Bayesian inference approach that was developed by Carl Friston, they tested different architectures, you can see here, different connections, and different modulatory configurations, which connections would be most important to get output of this model that is similar to output recorded in rats, anesthetized rats, which emulate Parkinsonian states, and the data is electroencephalographic or echocorticograms from those animals. So they had as real data the electrocorticograms of anesthetized rats that mimic Parkinson's state. They are animal models for Parkinson. And they adjusted the parameters of the model using Bayesian inference to choose the best architecture and and pinpoint the connections that would cause that increment in beta power. And they've got to this architecture, and they selected those connections here highlighted in dark red as the main ones. So this is the outcome of this work. And Ferrocchini and Lowery, the two guys who constructed the model that Raphael re-implemented, constructed a population model. It's important to say this is a population model like the Wilson-Cohen. It's a rate-based model. And what they did was to construct a thalamocortical model substituting the population model, which is just a single variable but by neurons. They had 540 point neurons, all of them of the Zikevich type, so that simplified model that I mentioned to you before, connected in this way. Although the neurons in each cortical layer are excitatory, they had self-inhibition, and this was important to provide a kind of inhibitory activity, intrinsically generated inhibitory activity there. What else? So they had all those. They used Sodexmarken short-term plasticity model, and they simulated a Parkinsonian state by altering the synaptic weights of the model to reproduce, to generate a model that has a high beta power in the spectrum of the local field potential generated. And then they simulated DBS pulses applied to the deep cortical cell, simulated that antidromic pathway that I mentioned to you. So what Raphael did, I'm getting close to the end, sorry, so I have time for questions. He re-implemented the model. Here are his results. So the model was connected in following from each connections between each population, each possible population, auto-all, so say a given neuron from the middle cortical column would send connections to, say the middle is connected, say to the inhibitory one, so CI here. So to all inhibitory cells, and all inhibitory cells also would be connected to all middle cell connections via with synapses that are random numbers between 0 and 1 multiplied by the maximum possible value. So each synapse would be a number between, say, 0 and the maximum value, randomly chosen. And what this diagram here shows to you is the average value, the mean normalized synaptic weights for each possible connection from always from row to column. You see here in red excitatory ones, say mean excitatory synapses, mean synaptic weights are excitatory, positive, and the blue ones are mean synaptic weights, negative. And highlighted in green here are the synaptic weights that are increased to generate a Parkinsonian state. So this is for a Parkinsonian state compared to the default, to the normal physiological state. And the blue, the ones highlighted in blue are the ones which are decreased compared to a default, to a physiological state. And by changing those weights, one would be able to emulate a Parkinsonian state. And if you compare this diagram with the one from the article by Reyes et al. That I mentioned before from the group of Freestone, you see that they are the same connections. So connections from the superior to middle, increased, and so on. So all of them, all of the ones who are highlighted here, show here in those strong arrows, with exceptions of the connection from the inhibitory neurons to the deep ones, which are not present in the model by Reyes et al., but were present in the model by Ferrocchini and Lowery, and which we implemented in our model, in our version of the model. This is the C to D, this one here. Okay, so but then that's the model. Here, so for you to see raster plots of the model. I'm sorry, it's in Portuguese because I took it directly from his TCC, the work that he made for his undergraduate diploma. This is the normal condition, no high beta power. This is the Parkinsonian condition. And then this is the simulated DBS condition. The current that simulated the deep brain stimulation is just a current that was applied to the right-hand side of the model, as I showed to you, like in the previous slides when I talked about single neural models, which reproduced currents with this amplitude. It's arbitrary units because the easy cavity model is a-dimensional, is non-dimensional, with a frequency of 130 hertz, which is the same one that was used in the original model. And it was applied to 10% of the neurons in the deep layer during 1 second here. And that's the result in terms of raster. And that's the power spectrum of the simulated local field potential. The local field potential consider only the synaptic currents, because the synaptic currents are believed to be the ones that originate this extracellular potential, this local field potential. Synaptic currents of the deep cortical population, excitatory and inhibitory. These are the parameters, and as you can see here, this is data simulated by Raphael. The physiological condition doesn't have peak in the beta. The Parkinsonian one has a pronounced peak here in the beta band, and the one with PD plus DBS has a reduced power. So it shows that the simulation, this in silico simulation, shows that indeed the application of this high frequency DBS to the deep cortical layer reduces the beta power. This is a statistical analysis of the model to show that's not only one simulation, but this is for, I believe, 50 simulations. This is the case for normal case, Parkinsonian case, and Parkinsonian plus DBS. It's significantly different from the case with Parkinson, and also different from the normal case, the physiological case. If you want to know more details, please talk to Raphael on Wednesday. And next steps. Well, this is his undergraduate project. For his master's, now TMS. The idea is to use the model, an extended version of the model, which now is our own model, is not based on a third-person model. Will be a model of the cortex, but without that common inhibitory input, because this is not really plausible. Inhibitory cells are present in each layer, so one would, in our case, we'll have each cortical layer having their own excitatory and inhibitory cells of different cell types, according to all the models that have uncovered the cell types that belong to each layer in the primary motor cortex with their connections. So we'll implement that as well, extending the M1 model and couple the M1 model to a model of the basal ganglia. I'm sorry, it's in Portuguese, but you can recognize it. This is the basal ganglia. So we plan to implement the entire circuitry of the cortical thalamic basal ganglia loop, and using not easy cavity, but ADEX, which is basically the same, but this will make the model different from the original one. And instead of considering DBS, we'll consider TMS, because we want to collaborate with groups from BAF and his collaborators as well. So the idea would be to apply TMS pulses, not to the deep cortical layer, but to the superficial one. Could be single phase, biphasic, and following all the procedures as well that I used to try and understand the effect of those into the activity of the layers, and also and see what would be the effect on the beta power, because that's the main signature of Parkinsonian state. So to conclude, so the thalamic cortical loop model, it reproduces the pathological beta rhythms in Parkinson's disease. This is one of the things that one expects out of a model that it reproduces the behavior which is observed. It supports the hypothesis that the connectivity of the thalamocortical circuitry is affected by DBS and affects the beta enhancement. Supports also the role of the antidromic cortical activation in the system, and shows that computational models are important tools for understanding how brain stimulation therapies, DBS and TMS, reshape the population level neuron dynamics. So hopefully this kind of model would help us to understand this kind of conceptual framework that Risto shown to us before, when the system jumps from one state to another one. Okay, that's it. Thank you.
Okay. Hello. Time for questions.
Thank you, Rod. I think the signals we can detect from electrical measurements or magnetic measurements are produced by thousands of neurons. And if I notice it correctly, your model simulates like 500. So what's in between this gap? So you think 500 is enough to get a reasonable understand of what's going on? And the other question is that we know that now the signal that's produced by the action potential is the summation of not a linear or a continuous current conductivity, but it's like the channels open, so we have like a discrete pulse currents. And you can simulate like the axon by just opening this discrete pulses. And since you are working computing, I don't know if this will, I don't know you add more complexity, but we would explain better the results if you consider now that the action potential is not a continuous function, but it is a summation of channels opening in a discrete way, statistically modeled.
Well, to answer your first question, this model that has 540, is not a neuron to model neuron. It doesn't have a mapping, a perfect mapping between a given neuron and a given simulated neuron. When you say that you have 500 point neurons, each neuron in that model captures the behavior of several neurons, hundreds, thousands of neurons. So it captures the collective behavior of given neurons in a given place in the cortex. Yeah. So no model. You have to understand the terminology. We say that we are using spiking neural models and point neurons, but we're not saying that each point neuron there has a counter part in the brain, because that would be really unfeasible. We have millions of neurons in that system, so or hundreds. Of course, one can. There are models that have this that are of this scale, but then we will need say supercomputer to do that, and we don't have this here. Maybe via collaborations we'd be able to simulate that. So that's the first question. The second question, I don't know whether I understood it perfectly, but of course the underlying mechanism of the generation of the action potentials is the random opening and closing of the ionic channels. But the model, even the Hodgkin-Huxley model, it doesn't consider that. It's a continuous time model. So it's a kind of average. There's a famous picture in the Hill book that's similar to the one that Risto shown to us. It shows the random opening of the ionic channels. But then if you add, say, yeah, you have a continuous function. So you can use, say, a continuous time continuous function to simulate this behavior. And then you have the separation between the detailed ones and the simplified ones. I'm here in the realm of simplified models, so just as two-dimensional variables, no explicit ionic channels, no explicit conductances, but the behavior is similar to the behavior observed experimentally. So that's the thing. Models, all models are wrong. Of course, this model is wrong, but some models are useful. We'll try to construct a model that can be useful for people who are really doing experiments and to understanding, to have insights at least to understand the phenomenon.
Okay, thank you, Professor. I have a question. I'm sorry if this is a simple question, but in a real brain, we have cells with different orientations. And can you model these different orientations of the cells because this is important in TMS when you create an electric field, and this interaction between this field and the cells are different because of this orientation. So can you model this? Is it simple to do?
I'd say yes, because since you have complete power on your model, you can say tag, say out of the say n neurons that you want to simulate, could be 500, 1,000, 10,000. You tag them, so this fraction of neurons have this orientation, and another fraction of them have a different orientation, and you can make this orientation a continuous variable. So you can simulate that if you want. And then you can study the behavior, the interconnection between them, without having a detailed compartmental model that captures the actual spatial extent. Of course, you can do that as well, but then it would be a completely different model. You can have models where neurons are really oriented in space in a three-dimensional grid, but this would be really more computational expensive.
Thank you very much. Very nice talk. So especially about that, like this kind of intrinsic properties of neurons there, how they are aligned, and how this information will change, how the activation of the neurons go. So we need to create somehow a bridge between these models and how these are going to apply in, for example, a clinical session in which you are going to put the coil on top of the head of someone, use the output of these models to decide how you are going to apply the sequence of treatments for that specific person. So my question is about how long does it take to generate this in terms of computational time, and like how kind of slow or fast is it? And how do you see this could be ported to this kind of clinical setting, real-time application, almost in a similar way as we have for radiotherapy, that you have the planning software where you have very good physical models of how the radiation interact with matter, with biological matter, and then you can kind of estimate those and things like that. So how could we bridge these neurophysiological neuronal models to kind of TMS applications?
Well, it is feasible, but I mean, it will take time. To start with, it takes, to simulate this model, please correct me, Rafael, but see, I guess a couple of hours would be okay, in a computer like mine. Yeah. So you can simulate in a very short time this kind of model. But of course, this type of model is very, very far from this, say, digital twin that I don't know whether this is the correct term, but that's the idea. We want to have a computational twin of a human person. So people are doing this, but in centers where they have large computational powers and they work closely together with the experimentalists. Here we are just starting. As I said, I never worked with TMS or even with DBS. This is the first work that we did. So we're really far here in Ribeirão Preto from implementing this. But I believe in other centers, it's possible to say via gathering data from different techniques, fMRI, electrophysiology, and so on, you can reconstruct a faithful digital twin of a human person, and then a human being, and then of the brain of at least of a given area of a person, and then try to simulate that. But for here in Ribeirão Preto, we're really far from achieving this.
If you allow me, just a comment about
Yeah, of course.
Your intention about this model is your patient are there, you're trying to apply the model exactly the same way that you're doing with the tractography, something like that. Yeah. So what's the input for the network to calculate what's the best orientation? Is the MRI, what are you applying to calculate it? Because calculating the weight using in patients?
Well, that's something to be defined, right? That's what we need to stop and think. But like just quickly thinking about it would be something like, if you have a very simplified model, of how different cortical locations interact, and then if you have like a certain neuronal population here and a neuronal population there, think about the depression network. One here, for example. Then if you stimulate here, what would happen with the interaction between these two if you apply a certain type of protocol? Is it going to work for that person or not? Can you adjust this treatment in real time such that you can actually increase the connectivity between these two functions based on neuronal models? You know, you cannot of course create neuronal models for each person, but you can simplify that based on where the TMS is actually stimulating, how intensity, orientations, and frequencies and stuff, and if that's going to be reflected for that anatomical organization in functional improvement. That would be how I would define it.
But it doesn't need to be online, right?
If it's online, it's better, no?
Like the radiotherapy.
Victor mentioned radiotherapy. It's not as simple as he think it is, right? It's getting more and more complicated with new techniques like flash, high dose rate, and so.
Okay, Professor Garrido.
Thank you, Professor Hoppe, by the nice talk. Very, all the time I learn about your talk. And my question is in one specific direction in terms of energy. I made this question before to you because I'm concerned in all this model, because in all this model you have neurons and connections, but neuron connection and all this stuff, but all this propagation of this needs more energy. So we are in an environment, and I have a curiosity about in this model, do you need to consider any restriction of the total energy of the system or something like this? Again, because in the brain, we have an energy consumption in different places. That is one part of the question. The second part of the question, what do you think when we are in terms of to model, for example, oxygen consumption inside the brain? Because I'm trying to do the linking between the macroscopic measurements that we have and also these kind of models. That is the two parts. That if you have any energy restriction in your models, this is possible, or and the other part is it's possible to simulate this kind of oxygen consumption or something like this. Thank you.
Well, in this type of model, energy is not an issue in principle. When it's trying to simulate, say, what is happening, say, during a given task. You have a experimental protocol in a real thing. You take an animal or a patient, you put them in, say, a given machine or fixing a screen and then do some experiment there. There's a task, the animal does the task, and then you measure things, and then you do statistical analysis. I guess even in the real case, people don't, I mean, of course, the animal is fed during the, or before or after, but during the test, they are concentrated on the elements of the experiment, on the activity of the cells that are recorded, and I imagine they at least in the papers, we don't see mentions about the energy consumption of the cells during that task. So the same thing happens here. We assume that the model is capturing the normal functioning of the circuit during, say, seconds or minutes, not more than that. So for that kind of simulation, energy consumption is I believe, irrelevant. Of course, it's there, but it's underlying. So, and to your second question, there's always the possibility, since this is a model, you can put in the model as many elements you want. So if you want to put oxygen consumption. You can couple to your model all the biochemical reactions that occur in there. The only thing is that your model will be more complicated. You have more variables, more parameters to set in order to have a simulation that captures the real thing. But you can do that, of course. Again, most of you are physicists, so you're aware about modeling, what people want to do and model. They, as I said in the beginning, want to simplify things, to try to understand, say, the principles. So I think the main take-home message out of this is that the cortical processing there has a contribution to the reduction in the beta power. This is something that is enough, say, for an experimentalist or someone who is interested in understanding better. Go there in the animal and try to, let's see if we cut this connection or we inhibit that one. Let's see what happens. So models are useful in that sense. They suggest possible mechanisms that can are candidates for implementing a given behavior. And then the task of experimentalists is try and then provide new data that the theoreticians test and so on. So there's a circle that moves science. So that's in that sense, that I propose that you accept this type of modeling. So I think he's still.
In terms of vascular disease, for example, in the case of vascular disease, definitely you probably have this kind of energy problems. This is why I asked.
But then you can, yeah, if you want to understand the role of vascular disease, you have to put in your model some element that simulates that. Lack of supply, energy supply to a given area, for example. Or you simply can say, due to vascular disease problem, I'm going to kill these neurons in that module, and then let's see what happens. Or introduce a dynamic to that. Or there's some period in which it doesn't have oxygen, and then it recovers later. So, I mean, you can do that, if that's your question.
I would just like to kind of upgrade to this conversation here. So you said in one of the first slides that basically model is accumulation of knowledge that we got so far, and then right now you talk about many modalities that we have data from, and then we are trying to link all these results from different modalities to kind of replenish or re-inform the models in order to calculate better. Now, my question is like, I don't know anything about this, but have you tried to use the machine learning or some kind of AI systems to integrate all these other modalities of the data and kind of extract the essential data that would be most useful for feeding your model? And then on the model results, to actually try to determine which of the models is most plausible to reality?
Well, I haven't. I have used with my students machine learning to choose the right set of parameters. So I want to implement a given model that does something, and then we, out of the, say, the sea of parameters, we want to find the optimal ones. We do this, and many other people do that as well. But try to select. I believe there are people using machine learning. As I mentioned, in the case of the Carl Friston group, they use those Bayesian techniques to select, say, the most probable, elements of the model that generate as a posteriori signal in the system out of the a priori ones that they consider the best candidates. People are doing, which essentially is the same thing, is do regression analysis, because machine learning is statistical learning theory in a sense. So you apply those, you can apply those techniques and select the best model. I don't do this, but it's done in other groups. So yeah, my answer is it's possible to do this, to select the best model.
Okay, thanks for your wonderful talk. I was a little bit uneasy about, not because of the talk, but you mentioned the post-Newtonian neuroscience or modeling or something. So do you have an idea what it would mean to be in the post-Newtonian era? I was thinking about, let's say, modeling earthquakes, We know the physics, the materials, everything, but we cannot predict earthquakes. Are we in that kind of situation that we understand everything, but cannot model correctly and predict how the brain works, Parkinson's disease or whatever? Or is it something else? What is really the post-Newtonian thinking here?
The simple question is, I don't know. But what I meant when I said that is that at the moment, I don't know about, say, geology, but in the area of theoretical neuroscience, this is my personal experience. I see many different modelers, famous ones, each one defending his or her own model. They don't try to put them together and construct a single model that can be used to understand the brain. That's what I meant when I say that we are in this pre-Newtonian stage.
Does it matter if your model is the right one?
Yeah, but each one believes that their model is the right one. That's the thing. And they went to, and you know how science works. They have the friends, so they publish the papers, they cite themselves in the journals, and they have a life out of this. But the real idea of, the real goal, which is to have, say, a model that can be used, is not achieved so far. So, but there are attempts. There are people who are like those simulators that I mentioned, NetPine, which is for detailed models, and Nest. People they're trying to construct a community of people who use those, say, those detailed models and add. So each research in different part of the world and in different part of the world add elements to the model, and then you build up a large-scale model that can be used, say, for single patient understanding or energy consumption and so on. Because only by, say, building up, using knowledge from different researchers and putting them together into, say, a given simulation, which could be placed in any place in Finland or in Brazil, a large computer could simulate that. Using those knowledges, we will be able to, I believe, in the future, be capable of predicting things about the brain, as well as about geology as well. And that's the thing about complex systems. It's both the Earth or the environment or the brain are complex systems. I think only by using, with help of machine learning, large-scale simulations, computer simulations of those systems, one will be able to get some understanding of the emergent properties out of that.
Okay, let's say that we will get artificial general intelligence and put all the data that is there in the internet, all the papers, and it then can predict. But we don't understand how it works. So would that be post-Newtonian or not?
No, it's not. I mean, maybe it's the wrong framing when I said post-Newtonian. It's my fault. Physics probably has a different story than, say, complex systems. By the way, we understand simple problems in physics, but the complex ones are really far from understanding, say, in that way. But anyway, Newtonian framework and all the frameworks that were developed by, say, Marx and so on, were able to make physics a distinguished area in the set of sciences, because we are able to understand very well many complicated problems. But other areas like the brain, the earth, or the environment, or say, pathology of the human body, are really so complicated that maybe thinking in terms of what happened historically in physics is not helpful for us to predict how will be the evolution of those areas. So my example at the beginning of my talk probably is wrong. I only used it for you to understand what I meant, but it's not that I believe that. I know history sufficiently well, or don't know, but to understand that things may take a different pathway than the ones that happened before us.
Okay, any other questions? Good. Okay, so we're back at 2 p.m. Okay, thank you very much.
bensound.com. 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 bensound.com.
So it's a great pleasure to be here. So back to home. And today we will have the pleasure to see another guy from Ribeirão Preto that started here. We worked together for several years. And then he moved to Finland with Professor Risto. And now he's a research fellow there. And he's developing a very interesting studies with TMS and multilocus TMS. So thank you. And Vitor, please.
Thank you very much, Andre. And yeah, I have to say it's just like the biggest honor I could have to be here today with all of you, because basically all my mentors and all the people that had a strong influence in my scientific career are here today. So that's great to be able to discuss these new things that we are doing and bringing science forward with this such amazing group of people. So I was also, like Risto said, I was also given this title, and Multilocus TMS, its beauty and challenge. I will try to give an overview of some of our recent developments there in Aalto University, and also in collaboration here with Bafo's group at this university. And try to also raise some of the challenges that we have to kind of solve, still to be able to kind of bring this technology to be widely adopted. Oh, what is a beautiful technology? But now I want to hear from you. What do you consider it to be a beautiful technology? iPhone. iPhone is a beautiful. Why? Okay. Yeah.
The developer is happy with the sound.
Okay.
Really, truly.
Okay, that's a good one. Yeah. So self-appreciation of the work.
Easy to use.
Easy of use. Yeah. That's a great one.
Works well.
Coffee machines. Yeah, they are beautiful. You press a button, comes the kind of pleasure in liquid form. Okay, these are good examples, and like most of the time when we're developing technologies, and like we kind of try to combine all of these, make people use something that we have developed so that it's easy to use to others, as well. If it's only you that can use it, then it becomes very restricted. It also have to look nice, kind of have this aesthetically pleasant look. So it's really, really nice when you kind of develop something and people come and say like, hey, this looks beautiful where it is and how it looks like. And so this is also important for patients, right? So when patients are going through a hard phase and then they go to treat in the hospital is already a heavy environment. So being aesthetically kind of comfortable and pleasant may also raise this kind of good feelings when they are being treated, and also kind of bring trust to the technology. So that's one of the reasons why we should make medical devices beautiful as well. They don't need to be ugly. It's like that thing of the electric cars. They don't need to be ugly as they used to be. So that was one of the big changes there. So I will stick to my analogy, even though Bafa was a bit critical about it, about the radiotherapy analogy. So let's suppose there's a patient, then there is a brain tumor in that patient, and then medical doctors decide that, okay, we need to remove those, or we need to treat that because it's gonna be really bad for the person. So then let's say that they decide that radiotherapy is one of the treatments, or kind of there's gonna be surgery, and then after that, radiotherapy. How does that work? Some physical biological modeling is used to compute the dose in a software. So there's a planning software that combines this information, like anatomy plus this physical modeling of how the radiation spreads. So then a medical physicist, together with medical doctors and technicians and so on, they decide, okay, like let's modify the treatment plan to save some tissue, or to bring higher doses to some tissue. And then, more or less, they put the patient there and more or less press a button, and everything works by itself, right? So there is a robot here that moves the kind of the gantry around the patient and delivers the certain amount of radiation in certain specific locations. And I think that's one of the best examples of automation in healthcare, right? Because neither the physicist or technician, they need to actually put there, oh, I need it to be in angle 35 and so on. There's a kind of a calculation of where these things go. So TMS is pretty far from that. Not so much with our technology now, but that's what we're trying to aim at. But that's not how it's been used so far. Just to stick to the also the comparison in terms of technological development, this is one of the first Macintosh computers in the 80s, in the early 80s, and this is how like a Mac looked like right now. Everybody have a pretty intuitive idea of how big development in this has been. TMS, so this is a photo of the 90s, but basically the first coils in the early 80s, also mid-80s, 1980s, they look like this. And this is how the coils look like in the market right now. So this one is, for example, McVenture announced this coil, T65, this year, and it looks basically the same as it was before. So technologically speaking, is just the same thing that has been for almost 40 years. Then comes the question, is TMS beautiful enough to be used as a kind of a treatment tool for the patients? So this is how an advanced TMS system in the market look like. So you have a person holding the coil, move around, give a pulse, measure the MEPs, move to a different location, measure the responses in the muscle, and then looking at the screen, there is a visualization of the brain there. So that looks nice already. It's pretty good technology. So a conventional TMS setup looks like this, where we have, let me see here. Yep. So where we have here a navigation system that guides the placement of the coil. There's an operator that places the coil around, and a camera that sees or tracks where the coil is relative to the head, and you can also monitor electrophysiological responses. So the problem here is that, well, sometimes in many cases, the brain function or brain organization has some millimeter, sub-millimeter level changes. So if you move millimeter by millimeter, you see different responses from the brain. And with this method, we have pretty much higher spatial resolution, I would say, than that. Then it's pretty slow. As you saw from the video, the person moving with the hand takes several seconds to move from one point to the other. Depends a lot on the operator, because the person needs to hold the coil. So the operator needs to be very experienced to make a good session. And it's basically blind to all of the intrinsic properties of the brain, such as connectivity. Robots come to help in the automation of this process. So this is a robotic TMS system that helps the placement of the coil. So it doesn't do many things automatically. Basically, the only thing that it does is just to hold the coil in place when the person moves the head. But robots have been used quite heavily to increase the precision of surgical procedures. So they are good helpers in this application. If you put the robot in the scene, that's how you were going to have the setup. You basically can remove the operator from being next to the chair to have this kind of setup where it can be basically controlled remotely. So here we gain quite a lot in terms of independence from the operator and kind of faster procedures and so on. But that's not enough, so we should think about how to drastically improve all of these procedures. And first of all, the one should be about treating the brain as a network system where we have multiple locations that communicate, and then diseases are affected by these problems in communication. You want to automate the workflow such that you press a button and you don't actually need to be there checking where the coil is or everything. You just press a button and, like in the radiotherapy, things move and the treatment is delivered. And it needs to be pretty fast because brain signals happen at the millisecond level, so we don't have time enough to wait several seconds to come to the other side. Just as an example, why network targeting is important. So depression is pretty well known to be a network disorder, let's say, where you have imbalance between communication in different areas of the brain. There are several networks that have been associated with kind of problems in controlling mood, which would be related to depression. Then just as an example here, you have the reward network in green, and then you have the striatum and the frontal cortex communication. This is a very high level visualization, but then you can have an idea that there are multiple locations in the brain involved in mood control. Treatments for depression, for example, TMS are usually applied in a repetitive manner to one single location of this network, here to the dorsolateral prefrontal cortex. Electroconvulsive therapy is similar thing. DPS, they implant an electrode, but you can see that all of these are basically almost single site treatments. You have one point that is being targeted. But then there are antidepressants that are basically kind of blasting the whole brain with the drugs. They can be agent specific, but they are kind of organ-wide activation process. So with TMS, here is if we apply a single site TMS, here would be the idea where we can apply TMS to multiple locations in that network and basically have better engagement with this system. Yeah. So our work has been to address all these issues so that we can enable network targeting, automation, and a very operator independent system with robotics and the multi-locus TMS system. So how does that work now? Here we have the navigation system. We send the information to the robot. The robot positions next to the head of the subject and moves around to find the target alone, independently. And then the treatment can be applied when the coil is on the target, or if we want to map the activation, map the motor cortex, that can be done also sort of automatically. So multi-locus TMS is based on combining several coils together. Here we have 5 coils, that's our latest prototype, and we overlap these coils one on top of each other and put them in this kind of casing. And then that's how it looks like when it's on top of the subject's head. Each of these coils generate a certain electric field profile in the brain, and then like each of them has a different profile, and then you can apply the fields combined, like you apply a certain electric current to each of these coils, and then manipulate the distribution of the field. So it's just a linear composition of these fields. And then by changing the currents, you change the location where the focus is. This is a bit exaggerated, just for illustration purpose. But the main thing here is that you can change the location, change the distribution at millisecond level. Instead of changing by hand, you keep the coils fixed here, and then you can change just the field. And changing these currents is super fast. It can be done at the microsecond level. So then millisecond level activation, that's kind of what we want for engaging with kind of brain communication between different areas.
Just a quick question. What would be the problem of taking too long to change the location? What's the problem that you have to switch like really fast?
For example, here, if you have a communication between the dorsolateral prefrontal cortex and the prefrontal cortex here, no, not prefrontal cortex, so what would be precentral? What would be PC here? I don't know. But if you have, parietal cortex, sorry, yeah. So if you have these two areas, and it takes seconds for you to move from between one and another, usually the communication between these areas happen at the millisecond level, right? So orders like hundreds of milliseconds and so on. So you wouldn't be, it's like thinking about you're pushing the swing, you're pushing the swing. So you would be always in a wrong timing, or you don't know the timing that you would be kind of, it's a different time scale.
It's like matching the frequency of this.
Sort of, yeah. Yeah, that's the idea. Yeah, so this is an illustration how it looks like in the kind of realistic anatomy. So we have the coils fixed, and then we change the shape of the electric field just by manipulating the currents there. So take focus here on this orange part. Now I'm shifting the field to the more medial region just by changing the currents here. That's how it looks like. And this can be achieved with this coil set here. And then back to the center, going to the lateral direction along the precentral gyros here. Okay? By doing this kind of millimeter by millimeter, we can map the representation of muscles on the motor cortex, and like here is the motor evoked potential, so how big the muscle kind of the activation of the muscle is, and see like where the thumb, for example, is represented here. And this is extremely important, for example, in pre-surgical mapping, where you might have a tumor here, and then you have a motor-related brain area, and the surgeon needs to know where to cut to minimize the kind of post-operative, post-operation damage. But also, it allows us to study some kind of neuronal mechanisms, for example, inhibition and excitation mechanisms, by changing the properties of the stimulation at millisecond level. So here we have two pulses, one in one orientation and another in a different orientation. That's paired pulse paradigm. And that allows us to activate selectively different types of inhibitory or excitatory, facilitatory mechanisms there in the motor cortex. And what we can see here is that depending on the timing between these pulses, we have facilitation or suppression of the motor responses. And if we change the orientation, there's also some dependency, like for example here in the inhibitory, if you change the orientation of the conditioning pulse, you might have different neuronal populations, and then start to kind of create an understanding of how these populations are connected, which of them in the cortex are kind of activated. Well, I wanted to refine this slide, but on short notice, I had to put it here. Here, we can also, there was this discussion earlier in the session in the early morning, like about simulations and experimental work. So here we are actually matching simulation of neurons with electric fields from TMS. These are the red dots here. So you see this red profile here. When we change the orientation of the stimulus, we reduce the amplitude of the response from these neurons. And these white squares, what it is like, white axis crosses here, this is experimental work, MEPs that have been evoked from the hand. So we basically match pretty well the responses, the depolarization of the neurons in simulation with the experimental collection of motor responses. One beautiful thing about the MTMS is that we can start creating different modules of these coils, not only kind of for kind of upper regions, but also for studying cerebellar stimulation. So this is one example of very recent coil that we are developing there. So here we have like the different winding paths here and more round shaped. They are housed in this kind of ergonomic casing that can be placed here, just like the headrest of a chair. How the wires are placed there and how the casing looks like. This is our last prototype. And with that, we can basically manipulate over the cerebellum also the electric field location and orientation to kind of map also cerebellar activation, which is extremely important for studying motor control. We can build coils also for small animals. So this is a work for combining multi-locus TMS and MRI in rats in a 9.4 Tesla MRI. So here is an example of how these coils can be used with a rat, and this whole thing goes inside the bore of the MRI. This is like just a schematic illustration, and we can apply pulses in different orientations and see how the motor responses from the rats change also in animal models. Another very nice thing that you can do towards automation and augmenting the way we see the brain. So this is how conventionally we have been targeting TMS, right? We take a look at the grayscale map, but they have no idea about the traffic of information or anatomical connections or anything like that. So if we start enhancing our applications with this kind of information, for example, tractography, we can select a certain target in the brain, see how it is potentially connected to different areas, and decide where to stimulate based on the anatomical connectivity between these different areas. And that can be done in real time. So we incorporated this super fast tractography algorithm in the navigation software that we developed here in Vesalius. And then we can actually like position the coil and then see what are the potential connections that we see there. But this is like just an information of the anatomical connectivity. We still don't have the information of the electric field, which is basically the dose of TMS in the brain. And that we can also do in real time during the positioning. When we place the TMS coils over there, we can compute using realistic head models and electric field computation in these realistic models, how the electric field is distributed, so that we can also decide how to position the coils to minimize the stimulation of some neighbor areas, or to have it more focal or less focal, whatever is important there. Okay, so far, isn't it too good to be true that everything seems to be amazing? We can do network targeting, we can see in real time a lot of stuff, but it's not as simple as that. One of the main challenges that we have still to solve is to cover the whole brain. Because as I was showing in the previous slide, is here we have multiple areas that are kind of distantly located, or they are neighbor areas, and so on. With our current MTMS system, we can basically stimulate that area, change the stimulus within that red circle. So a next extremely important step for solving this whole brain network stimulation would be to have coverage, extended coverage. There are some other groups developing multi-coil systems and placing these coil modules here. For example, these are 3 coils wound in kind of different arrangements, and then they are placed inside these holes in a helmet. And then this can also be used inside the MRI. That's the target application of this group. But then they have these modules that can combining the electric field of these modules, they can shift the location between different locations or stimulate also different locations. It has a lot of problems, technical problems to be solved, but it's one approach to kind of have whole brain coverage. With our colleagues in Spain, we have been also thinking about some type of helmet for our multi-locus TMS arrangement. And here we would have like these segments spread around to be able to target specific regions in the brain. So that's some very early stage development that we have. Another one is that in clinics, TMS is almost only relevant in repetitive applications. There are also some diagnostic applications of single pulse, but repetitive protocols are the most used to treat depression, to treat OCD, substance abuse, and so on. Because we require a lot of current, like very high currents in very fast succession of pulses, then we need to have cooling, because the high current is going to heat up the wires, and then we need to cool them down as much as possible. Here you have this MagStim coil. That's a clinical FDA-approved system there in US. And it's just one coil, and it's this big, because it requires this lot of space for the cooling unit, for having the fans here, and so on, just for one coil. And now when you look at our system where we have these overlapping coils, it's very difficult to incorporate cooling here. So we need a smart arrangement or a smart mechanism that we still need to figure out to be able to cool down these, because they heat up pretty fast. So that's one big challenge that we still need to solve. Another one is kind of portability. So that's how our setup looks like with the power electronics. That's pretty big. Then there's the robotic arm. That's also big. There's also the computers and the EEG, EMG system. So some of you already visited the lab. You saw there like how these components are built together. So here we are using a system that needs to be set in a room, and then the patient needs to come. But in many applications, for example, in depression treatments, might be relevant to think about home-based applications. There are a lot of efforts trying to develop this. For example, one that recently came out as this almost look like a movie kind of device. It's like a backpack that has the coil attached to the head. This also looks too good to be true. So something needs to be evaluated here. And just still keeping the connection with the previous talks here, we have to still incorporate much better like the physical and biological models to kind of estimate the dose and estimate like potential activation, so that we can be more effective for individualized protocols, so protocols that can be adjusted to each patient. So there are also some efforts in kind of trying to compute the electric field in the brain and then compute the electric field on the neuronal kind of arbor dendrites and so on, to estimate the depolarization of the main brain and see like, okay, what is the actual physical dose or neurophysiological effect and the depolarization in each of these neuronal segments, so that we can kind of infer and utilize this information later on. So this is, as we discussed also, this is a bit difficult to think about in how we would implement that in a TMS workflow, but it's something for us to consider as a next step also, if we want to be more accurate. I didn't have time to update the thank you slide, so if your name is not here, please don't be, this was from another presentation. But yeah, so thank you very much. This has been like a big project that has been developed through a lot of people in our lab there, here at the University of São Paulo, also at the Center for Technology of Information in Campinas. So it's a big, big effort from a lot of people. So thank you very much.
Thank you, Victor. Hello. Thank you, Victor, for your amazing talk. So very impressive technology. And now we have, if you have questions. Sir.
Victor, you mentioned the concept of dose. And this is something that's kind of haunting me because when you make a comparison with radiotherapy, so going back to radiotherapy, when you talk about dose, you are measuring the energy that's deposited in the tissue divided by the mass of the tissue. So this is the macroscopic concept of dose. But it's some sort of energy that you deposit and the amount of mass you have there. But then when you go to brain stimulation, you see all sorts of definitions, right? So I was in this conference in Kobe, first time I attended, amazing conference. So I was watching all talks, and then you see people that working with direct current stimulation. So they put 2 electrodes and they discharge a battery, and what they call dose there is the charge they deposit. But even that, when you think about it, so you are injecting charge in one side and getting back from the other side. So I got confused how I can associate the dose with the charge of the battery. And then you see those guys that are working with ultrasound. So they put some wave pressure, something vibrates the main brain. You know, the mechanism is still under debate to be fully understand. But they talk about dose also. And then we talk about TMS. So we apply a pulse. Risto did show that in the neuron, we have a balance of charge, right? Discharge and then charge and recharge. So the integral of, let's say, the going up and going down, then should be balanced, right? Same thing you have with MRI. I don't know if Garrido is here. So I think this is a very important concept that needs to be somehow worked out so that we can compare different techniques. Because right now, I think when you say dose, you can say in the context of TMS or in TDSS or in ultrasound, but they are not interchangeable, right? You have to somehow think about that. So what's your take on this problem?
That's a really tough one because it's really poorly defined in the field. It started with for a very long time, the main indicator of dose in TMS has been like the percentage of the stimulator output. So it's basically like how much you have turned the knob of the system to define the intensity of the pulse. I don't know. I think, as you said, a really big effort needs to be put into defining. Like, first of all, some good measure or some good approximation of the electric field is the first step. And that then combined with the neuronal depolarization measure somehow, you know, like the amount of electric field that you put over time, that results, because the time here is extremely important, right? How much, it's not only the intensity of the electric field. So some systems just show now the electric field as an intensity measure, like, oh, it's 50 volts per meter. But that alone also doesn't mean anything. You need to have electric field over time, because that's what will cause the depolarization, the action potentials to be generated. Oh, kind of, I think there needs to be kind of some physical understanding to present a certain measure that is meaningful. for real and try to simplify less, a little bit less than what it is. Risto might know a bit better how to explain that. Yeah.
No, you explained it very well, but one aspect is how many pulses you are given. So if you have, in a typical treatment, you can have tens of thousands of pulses, and it's well defined that if you double the dose, it's twice as many pulses, right? But then the individual pulse is more complicated because it depends also where, as you explained, the duration of the electric field and the area that is stimulated or the volume. It's different in the different devices. And
there's
some are more focal, some are less focal.
Yeah, there's also like with radiation, for example, there it's a cumulative effect, right? The longer you expose it, the more energy you deposit, and so on. And then with TMS, if you want to actually kind of enhance the connectivity between neurons, or if you want to kind of promote plasticity or kind of use plasticity at your benefit, then you also need to kind of see at the long term with these many pulses, how did it accumulate over time? And that's really hard to kind of, I don't know. But
well,
at least use these concepts wisely.
I'm receiving a call from my insurance company. So I think it's a tough question, and perhaps we could somehow mimic what happened in radiation physics. Because when you talk, let's say, about radiotherapy, you can have ionized radiation of different kinds. So you can talk about, let's say, photons, charged particles, and heavy charged particles and light charged particles. And every one of those modalities has a completely different formulas of interaction. But somehow we manage it to establish a common ground where we can compare. Yeah. So if a physician prescribed that a tumor needs to receive 10 gray, it's that you know what's the dose, but then you can decide, well, I can give 10 gray with photons, but photons will spread around the tumor. But if you are in a very sensitive area, let's say in the eye, and you want to spare the eye, okay, let's use protons, because then you use the Bragg peak, and then you just locate in the right position. So I think it's the same thing here. We should put some effort on that.
More questions?
Can I make just a comment before? Yeah,
sure.
Just one small comment. I would like to encourage also the students to make questions, because sometimes we are kind of kicking off the discussion here. Don't be shy, and also if you're not comfortable with making the question in English, for example, just make it in Portuguese and we translate. Just engage in the conversation, whatever question it is. These are complicated topics that took me kind of tens of years to barely understand. So,
Thank you, Vitor, for the presentation. It's a beautiful technology and also a beautiful presentation.
Thanks.
Too. I would like to ask you about one of your results. You implemented the tractography to the navigation system, so you can see from the side that you are simulating which are the connections, okay? But are you able to verify that by stimulating that site, you can get a response on the connection that it's presented or not?
Yes. One example of that is combining real-time tractography with TMS and EEG. Because with EEG, you can see at the millisecond scale that the action potentials are propagated, have propagated some other side. So there are some pilot studies that have been done in the lab there by our colleague Pantelis Liumis, for example, that give a pulse here and then see that action potentials are that evoked potentials can be seen on the other side, but not when you move the target to a site that the tractography doesn't show any connections to the other side. So you look at the tractography set, put the coil more or less on top of that, it goes to the other side, nice, you get evoked potentials on the other side. He has done it for the prefrontal cortex. And then, like, if you move to a target that doesn't show these connections, then you don't see those evoked potentials on the other side. So there can be kind of this confirmation, validation of the idea. Then TMS EEG is good for that.
Thank you, Victor. It was a pleasure for us also to receive the whole system and set it up here in the lab. You didn't show the interface of the system where you can choose the exact point. So in millimeters, you have a square with 30 points across X and Y, and you can choose millimeter by millimeter where you want to go, right?
Yeah.
My question is, would we gain from defining a minimal distance that we go to the site to translate that pulse, and then for that person, and see that we have another different kind of configuration, a different kind of consequence of that pulse? Probably 1 millimeter is not enough to have a different thing, but we are mapping the motor cortex. We need at least 2 or 3 millimeters since until we see a difference. Do you think it's going to be a kind of personal thing, or it's going to be the same for almost everybody?
No, I think it's very personal thing, especially because the representation in the motor cortex is heavily dependent on the types of activity that you perform. So it is, my assumption is that it is very different between individuals. But like one thing that I also didn't put here is that there is an ongoing development in the lab to automate this whole mapping procedure. So ideally, you would just use it as a black box. You don't need to know how many millimeters you need to shift. You press a button, and like the system kind of probes different locations, and then based on some model that you do, then okay, the likely localization of this is here. So there are also other group in Leipzig that has been working with the localization, like by just giving random pulses in random orientations and using the electric field information to kind of compute back where it's mostly represented. It's not that simple. There are some recent studies on like the somatotopic organization of the motor cortex, that it's not really like the Penfield homunculus that you can see from the MagVenture stand over there, that it's much more kind of distributed in modules that are jumping, and that they work together to generate movement. So it's not like thumb is here, index finger is there, like it's more complex than that, as we would expect, right? So that kind of stuff. But I think it's individual and should be kind of established. Some work, some other don't, and that's it.
Let me make a question also. So I'm going back to the real-time tractography. It's amazing and beautiful. That's this. So we know that it's very hard to target deep structures in the brain using magnetic stimulation because of the characteristics of the magnetic field. But you have this tractography that, so in real time, so more or less you can target or you can see structures when you stimulate the external, so the cortex. You can see some deep structures, let's say, for instance, the hippocampus. And you can stimulate several sites simultaneously using your system, if you understood it right. So how do you see the possibility of using these tools together to try to stimulate or at least target deep brain structures?
Risto, you want to take that?
Well, to go to the deep targets, you select from this image. You look at the track. Some of them go to deep targets, so you can indirectly stimulate the deep targets by selecting a locus on the superficial cortex that goes down. With multi-locus technology, you can do even more. You can stimulate, let's say, 2 or more locations at the same time or short distances and short time distances, and which lead to the same deep target. And that allows one to study at this moment, because we do not have experience yet, spike timing dependent plasticity at depth, because you can give first pulse to the first location and then a couple of seconds later the other one, or vice versa, and see what is the difference in the result. Yeah.
I think that when that we have the tools, and I mean it's just about now defining the protocol, right? How, when, what timing, like what is the perfect adjustment of these settings. When that is done, it's gonna be like a super beautiful.
Yeah, so this is a game
result.
change.
Yeah, yeah. This
So-
change is the kind of, it brings TMS to deeper structure.
Have you tried with
No.
some pilot or something like
No,
that?
not yet. Yeah, I mean, like it has been discussed with. That's why I
Okay,
threw
great.
to Risto, because that has been discussed like heavily in the past years within our group, but somehow we didn't try it yet.
Great, thank you. More questions? Questions? No?
Oh, thank you, Victor. Do you think that the hotspot can be constant over during the experiment? Because I believe that there is one work from Risto's group that said that the hotspot can be not just one point, but one area. So do you think that using the multilocus TMS, we can learn more about the hotspots? Because until now, we just set one point and go ahead, entire experiment, and maybe it cannot be true. Do you think that?
I mean, like if it's an experiment within 2 hours and so on, like what we call it hotspot, which is like the location that evokes the maximum MEP amplitude, or the coil location, it's not even in the brain, right? It's the coil location on the scalp, right? No? It can be defined, like the definition of hotspot is as complicated the definition of those almost, but yeah.
No, it's not. I don't think it's complicated. But initially, when MRIs were not used, people defined the hotspot as the coil location. But now that we know where the E-field maximum is, or where your target is, so the tendency is now that people use the word hotspot as the location in the cortex from where the pathways go to, let's say, the spinal cord and the muscles.
Yeah. I mean, that's by a very selective group that has this kind of devices. Like most of the world doesn't have, and they still keep that definition that hotspot is the location where
the transformation is going on. Not,
is going on.
not
Yeah,
only
that's true.
with us, but others are starting to use it.
Yeah, yeah.
Real hotspot is in the brain. It's not
Yes,
somewhere
definitely.
outside the brain.
Yeah. So if it remains the same during the whole experiment, I think yes, like it could be that it remains the same, but also it can change depending on a lot of stuff. So for example, with Marco, we have been studying like different arm postures and so on, right? And that can affect like the whole cortical excitability. It's not only what is here, but it's all like, so it should remain there, but might change. Yeah.
Questions? So we have time for our two questions to do this.
Thank you, Victor. You showed image about pair pulsing TMS, so the dependency on the intensity and the orientation of the pulses, and also the time. But what I would like to ask you is, if in your experience, did you notice that the time is also individual dependent and the orientation? Because the excitation and inhibition is not always present, even with the same paradigm of pair pulse with different subjects. So
It's pretty consistent, I would say.
yeah.
Like especially inhibition. Inhibition is a very strong effect. Like so you really see it with these timings. They in this paper here, I have the supplementary materials there, the figure of the MEP amplitudes and orientation and everything for each subject. And like from 12 subjects, 10 of them had almost the same profile. Like it was really consistent. I mean, this has been studied quite a lot, this SICI and ICF. So like I made a bad choice of choosing 8 milliseconds, should have been something like 10, 12, which is a bit stronger effect. But I would say that it doesn't vary that much between subjects.
Yeah, I mean, the phenomenon is there, but I mean the proportion of inhibition and excitation.
Oh, yeah, definitely. That will change because it also depends on the kind of one person. If you give a same type of stimulus, some person is going to get 1 millivolt response and another person is going to get 200 microvolts response, or 0.2 and 1. And the inhibition is not going to bring them, like some of them just abolished the MEP. So for one, it's going to be like all 80% reduction. For others, it's going to be like 50% reduction. So I don't think that most of the studies in ICF, ICI, SICI and ICF have used like these relative measures, so normalized to the single pulse. I don't like that at all. I think that's removing a lot of the good information. So it should be analyzed in a kind of individual basis. In that sense, yes, definitely.
Thank
the
you.
proportion changes. Yeah.
More questions? No more? Okay, so thank you, Victor, for an amazing talk.
Thanks a lot. Now we have time for another breakfast. Oh, no, another coffee. Who watched The Lord of the Rings? You have the first breakfast and then the second breakfast. So it's Hobbit land. And let's go for the second breakfast.
bensound.com. 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 bensound.com.
Start again. So as you can see, we are bringing people from several parts of the world, I would say, right? To try to connect them. So it's not brain connection also, but research group connection. That's what this is all about. And now I have the pleasure of introducing you Professor Rodrigo Bazan. He's an associate professor at the University of State of São Paulo. So São Paulo has three main public universities, USP, the first one, UNESP, and then Unicamp. And he's from UNESP. That's about 300 kilometers from here, right? 200, okay. And so he works in stroke. He'll be talking about neuromodulation in this area. And we hope this is the first time we met personally. We had talked a lot over the phone, but we hope this is not the last one, but the beginning of, let's say, a fruitful collaboration among our groups. So I'd like to turn the word for him now. Thank you.
Bom, inicialmente agradecer ao convite aí do professor Oswaldo Bafa, né, para estar aqui hoje. Eu já peço desculpas de antemão que meu inglês ele é muito ruim, né? Algumas partes eu vou me arriscar aqui em respeito aos colegas aqui da língua inglesa, mas no geral algumas partes eu prefiro falar em português e para o debate depois, com certeza algum colega pode nos ajudar também fazendo a tradução. Bom, eu sou full professor em Botucatu, Unesp, né? Atualmente sou chefe do departamento de neurociências, né? E nós trabalhamos, né? A Fernanda Winkler está ali, a Luana é aluna de doutorado. Nós trabalhamos numa equipe que nós realizamos algumas pesquisas em neurociências. Lá nós não temos um laboratório, né, específico de neurociências, mas nós fazemos neurociências no hospital, né? E a gente meio que sobrevive dessa forma, se agregando ao hospital e aos departamentos e tentando ocupar um espaço. E agradeço novamente o convite. Então, a proposta da aula é falar de neuromodulação não invasiva em AVC, né? Então, é isso que a gente vai falar. Na verdade, basicamente vai ser um trial, né? O Titan trial, this is a second trial, né? Made for us in Botucatu, in neuroscience department. This trial is in the past, I've heard about the neuromodulation, non-invasive neuromodulation techniques in clinical practice in neuroscience and psychiatry, and I find very curious methods, and I and my team, local team, designed this second trial, Titan trial. The first trial used TDCS, the electric stimulation, is another technique, another device. In a specific Titan trial, we are using TMS and this sequence, specific sequence, theta burst sequence. Our model is a human model. In our field is the patients after stroke. 38%, 35% the patients after stroke presents signs of disattention or negligence, spatial negligence in the left side, and these patients is the target of this research. And the TMS and the TDCS plus TDCS, we are using this case. Stroke is a very common situation, clinical situation. It's very severe. Is the main cause of death in Brazil, and the incapacity too. O cabo aqui está desconectado? Acho que é o cabo, né? And a little problem of device. And in our hospital, student hospital, clinical hospital, we have a stroke unit, around 12 beds, and it's very frequently situation after stroke. These signs, the hemineglect, spatial, unilateral signs, very common after ischemic or hemorrhagic stroke in the right hemisphere, a man in the parietal region. Unilateral spatial neglect is a curious situation about the patient disperception of the environment, of the parts of the body. This deficit is very severe too, and associated a poor functional prognosis in putting these patients. And this many times this situation is not recognized for the clinicians, for the doctors, and it's very, very, very important situation. In neurorehabilitation techniques, we have many techniques and devices for treatment these patients. Neuromodulation is a technique, unusual technique for the specific situation after unilateral spatial neglect. And Pascal Leone is a researcher at the Harvard Medical School, and this researcher created and studied for many years plasticity, neural plasticity mechanisms, and the inhibition, interhemispheric mechanisms of inhibition, right and left hemisphere, and the relationships of about two hemispheres. When you cause a lesion, in a specific case in right parietal or right hemisphere after ischemic stroke, we have symptoms of disattention, paresias, and many other symptoms. And beginning a growing around this area, the zone of suffering, this ischemia, we have a plasticity, neural plasticity. In normal circumstances, these patients assisted for the team, of stroke team, physiotherapists, doctors, nursing, and many others, occupational therapists and the speech language therapists. And these patients, after discharge of hospital discharge, continuing keeping this rehabilitation, motor rehabilitation, sensitive rehabilitation, and cognitive rehabilitation in ambulance situations. This theory brings us about the plasticity and relationship with the lesion and another no lesion hemisphere. In this case, left hemisphere, no lesion hemisphere, and the relationships, the power of the inhibition in the left hemisphere about the right hemisphere, under right hemisphere, and these relationships. And this is the techniques about the neuromodulation invasive or no invasive, neuromodulations using TMS or TDCS and the others, vagal stimulation, nerve vagal stimulation too, bring about the open these mechanisms of plasticity, neural plasticity in the neurorehabilitation techniques and the other situations. This relationship is very complex. In general, the region with lesion no generates signs, electric signs. The brain generates electric signs, and this area metabolism is long, is depressive metabolism, and this bringing the symptoms, in this case, hemiplegia, hemiparesias, dysarthria, and no recognizing the left space of the environment or part of body. The principles about these techniques, inhibition or stimulation, disinhibition, these both hemispheres simultaneously or not, inhibition or disinhibition, in contest after a stroke, and observe what occur after these sessions, the neuromodulation sessions, in the space, in the time. In the first trial, electron trial, our results were positives, and results in publication in Annals of Neurology is a great paper in great journal of neurology. Think about this, decided keeping walking in this field. In this case, TMS in specific.
bensound.com.
in TMS techniques, this is a non-invasive neuromodulation technique using magnetic fields to induce focal electrical currents in the cerebral cortex. Depolarization, this network of cells or neurons is a very, very, very good. We have a network system, neurons, lesion area, body of neurons, and these prolongaments as depolarization in this network. And think about the plasticity, neuroplasticity is a good mechanism for the rehabilitation, neurorehabilitation, plus techniques, conventional techniques, consagrate techniques. We have physiotherapy techniques and the others. Now, these currents depolarize neurons in a controlled and safe manner, modulation cortical excitatory mechanisms. And when you use it, low frequency TMS, less than 1 hertz, this effect is inhibitory effect, and when you use this plus hertz, 1 hertz or plus, you have a excitatory effect in the cortex and subcortex of the brain. And this technique, in physiological effects in this technique, we have a modulation of synaptic network, excitatory network, and this influence on cortical and subcortical networks of the neurons, brain neurons. A frequency and duration depend plasticity, long-term potentiation, or long-term depression-like. And TMS in beginning applications, in medical applications, I imagine in psychiatric situations, the very common situations are depression, schizophrenic, and the other situations. In the field of the neurorehabilitation is still have a problematic situation. The potential risks, effects, side effects like crises and headaches and the other effects, in general, cause distance between the researchers, the medical group, and the patient using in these TMS techniques, after stroke. Mainly after stroke. Stroke is a situation very, very complicated situation. Many mechanisms are involved during stroke and after stroke. You have a great cortical reorganization, and thinking about neuromodulation in this cortical region is problematic, and the level of evidence and use this technique in clinical practice is very low too. And however, we decided we'll do this trial. I hope so, and go ahead. On theta burst stimulation, we decided for this type of stimulation. In the systematic revisions, showed for us about this technique, specific technique. In some cases of neurorehabilitation is a optimistic vision about theta burst stimulation. And we have decided that using this technique is a very short time when compared of the TMS, common TMS. In the after slide, you have seen about this. A variant TMS based on theta frequency patterns, 5 Hertz, delivering bursts of 3 pulses at 50 Hertz. Rapid induction of depolarization network regions induce a plastic synaptic. And this is a great situation. When we have a open plasticity for the shortly time for the sessions of the neurorehabilitation sessions, and hope find the good results in this the end of this trial. And the more efficient modulation of cortical and subcortical stability in less time. And the time is very important too. When used, the TBS continuous, the pulses are continuous pulse with effect are inhibitory, sounds affect inhibitory effects, and intermittent TBS excitatory effects. We expect the excitatory effects. And in this case, in this trial, our option for the inhibitory effects in the left parietal cortex, and for the inhibition, this parietal no lesion area of this cortex, left cortex, parietal cortex. About this, remember in the beginning of this lecture about the Pascal-Leone theory. When we have inhibitory of the left parietal cortex, we promoted the benefits and the I hope good results in the right cortex, and the good results in clinical, find clinical results too. And the rapid induction, when in the network area, in the right parietal cortex lesions, when talk about the side effects, in the reality, headache is a very common situation in TMS, and less common situation in TBS and theta burst stimulation. In general, TBS, theta burst, less discomfort about the shorter duration of the sessions. And the epileptic seizures, a very rare situation, mainly with right frequency stimulation and in at-risk patients. The large core or the large glios area after stroke, and the patient suffered the crisis during the acute phase of stroke, and in other situations too, or before stroke. In these patients, is better using the TMS techniques or other types of neuromodulations, invasive or non-invasive techniques. In general, headache is very common. In around 50% the patients talk about the headache during the sessions, and part of this need to stop at the session for discontinuing the session about cause of the headache sensation.
Hey, hey. Where do you think the headache comes from? It would be from scalp muscle contractions also leading to headache, or is it just like what's the origin?
Probably the combined mechanisms, the contrain of the muscle mass of the temporal muscle. Yes, we have this situation, and the headache is a complex mechanism in general, involved the trigger, the trigeminal zones, and the many others. As possible, when you stimulate the proximally of the gated of these regions of a headache, beginning of the headache, involved in headache, as possible, tension situations, muscle tension, and cervical. I think about the combined mechanisms. I don't remember read about this and the specific physiopathology in this case. It's very common situations. 50% is very high, and around 15% needs to stop at these sessions about this gravity of the symptoms, intensity of the symptoms, and the 2% don't back of the therapy about this. The scalp contraction, combined mechanisms, but it's very, very light situation. The main problem, like epileptic, is very, very rare. TDCS, used in the last trial, the electron trial, use the low intensity electrical current. Is a current applying the scalp, and this current 1 or 2 milliampere with electrodes in the scalp, and subthreshold modulation of neural stability. These mechanisms are very different compared to TMS. TMS uses the magnetic fields, and the TDCS use the continuous current or alternate currents, electric currents. In this case, probably we don't have a despolarization neural network, brain network, only a changing this environment around the neurons, and only this. No provokes real depolarization in the neuron network. Induces a prolonged neuroplasticity via mechanisms, many types of mechanisms. In general, the anode increases stability through the depolarization, cathode decreases excitability, and provokes hyperpolarization. This technique was used for us in the electron trial. Is the first trial used in neglect patients after a stroke, and the results were good. Neuronavigation for the localization in the real world in our center, in the many centers of Brazil, and the rest of the world too, used the 10, 12 system. This system is a very points, distance of the points very kinology, and this is a simple technique, is very quickly in the market on the scalp the patient, and is very used. However, the neuronavigation through these modern techniques of the neuronavigation or modern techniques, the image techniques are better, very better, and very expensive for many centers. And this in specific demonstrated for Vitor in the last session. It's a system allows precise localization of specific cortical regions in the real time during TMS or TBS application. Is the real dream for the research. When we compared these different types of techniques, TDCS, TBS, and TMS, a duration of the session is very important. The TBS, 3 minutes, in median 5 minutes of the duration of the session, and the mechanisms are very different mechanisms. TMS promotes a cortical excitation or inhibition, high inhibition situation through the depolarization cells, and the TBS is faster and longer lasting compared to TMS. And the TDCS is very, very simple technique, and it promotes a subthreshold modulation with the anode or cathode electrodes in the scalp, applied in the scalp. When we combine these techniques, use the simultaneously 2 neuromodulation techniques, enhance the recovery from unilateral spatial neglect after stroke. Is the good question. The question is great motivation of the Titan trial, and this technique is demonstrated during this lectures, during this meeting, and this is a Titan trial. Titan trial is a first trial. This trial used the double-blind randomized controlled trials with 15 intervention sessions around this over 5 weeks, and a follow-up period of the 6 months post-stroke, after stroke. 54 patients with early subacute ischemic stroke will be enrolled. Why subacute ischemic stroke? And the hemorrhagic stroke is very complicated. These patients present many problems, infection problems, and this incapacity is very, very aggressive in the subacute phase. And in general, the stroke, ischemic stroke, is better model for us applied this model of this trial. And these patients will be enrolled and during 15 weeks. So sorry, 56 months. This trial, many centers will participate in this trial, like Toronto, Vitor, Pereira, Western Hospital, Unesp, Ribeirão Preto, USP, and the other centers of Brazil, and the centers in the Australia too. Professor Brenton and Professor Emily will participate too. This inclusion criteria, both sex, age in 18 and 85 years, diagnosed with ischemic stroke, confirmed, presented in CT scan or MRI in acute phase, in the early subacute phase. After confirmation of the ischemic stroke, after 7 days after stroke, 2 to 3 months post-symptoms onset, these patients enrolled in this trial with right hemisphere lesions, now specific in cortical, parietal cortex, right cortex, as possible a subcortex or other areas in the right hemisphere. The main situation is the clinical situation of the unilateral spatial neglect, confirmed with lesions in the right hemisphere, in general in parietal right hemisphere, and they identified using the behavioral inattentional tests, named the BIT test, scoring under 129 points in this score. These patients will enroll in this trial, and we are randomizing in 3 groups. The exclusion criteria presents is a very, very long list. Epilepsy, for example, bilateral lesions, and many others, pregnancy, hemorrhagic stroke, clinical instability, difficulties of the communications like a global aphasia, very, very difficult of dysphagia, and many other situations, clinical situations. We are discharged this through the exclusion criteria, and the phase of screening. This trial no beginning screening, and these patients selected these patients for 3 different groups. Group 3 probably is a group on the group 1, group 2. We use it in the real time these devices. E placebo group probably is a group 3 on where you use these devices. However, these devices will be stopped in the name of the Shan group. In this model, group 1 uses in the left parietal cortex the inhibitory pulses through the TMS, or in this case TBS sessions, around 5 minutes, and the right hemisphere through the anode devices. The excitatory cortex, right cortex, lesion cortex, will be extended through the TDCS, and group 2 only TDCS, and group 3 Shan group.
Professor, just one question, please. Are you recording motor vocal potentials?
Motor?
The EMG? Vocês estão fazendo registro do potencial evocado?
A intenção seria fazer. Quando a gente fez o TDCS, não fez o registro. Não fizemos o registro. Agora, com o TMS e o tetabarcino, a gente gostaria de fazer. A gente não sabe, a gente estava conversando agora com o professor Bafa sobre isso, a gente não sabe se o recurso nosso vai ser possível, quanto de tecnologia que a gente vai ter envolvida aqui, a gente vai conseguir realmente fazer, que nem um neuronavegador, né? Ele custa mais caro que o que você vai fazer a estimulação, né? E realmente são aparelhos caros, depois do dólar, né? De tudo isso, é bem difícil, né? Seria o ideal, seria o ambiente ideal, mas provavelmente para o trial a gente não vai fazer, a gente vai pressupor só. Fala.
Tem uma pergunta relacionada a essa. Felipe.
I have a related question, which is, then if you don't measure the motor evoked potentials, how do you define the dosage, the intensity in the stimulator? Because generally, as is my understanding, you define the intensity of TBS in according with the individual resting motor threshold.
Ok, nesse trial não. Quer dizer, a gente não vai conseguir individualizar as doses. Você vai ter um tempo de estimulação, in the time, né, short period of the time, during around the 5 minutes, and the 30 minutes for the TDCS techniques. Falar em português, o mais importante não é só a neuromodulação. Todos os estudos que mostraram só neuromodulação falharam. Esse talvez seja o maior problema da técnica. Quando você pega e vê as revisões temáticas que tratam da neuromodulação não invasiva para neuroreabilitação, a maioria é negativa ou não consegue finalizar o que quer mostrar, justamente porque faltou, né, aquela coisa que hoje é muito importante em técnica de neuroreabilitação, que é a neuroreabilitação tem que ser focada à tarefa. Quer dizer, o indivíduo ele tem que estar exatamente como o colega mostrou, né, como o Vitor mostrou. Quer dizer, o paciente ele tem que estar com um dispositivo portátil e fazendo uma outra atividade. No caso, já existem atividades já que foram pré-determinadas que esse paciente vai fazer, tá, durante essa estimulação. Quer dizer, quando você faz a neuromodulação, é como se você abrisse um portão ali da neuromodulação, da plasticidade cerebral, e você, através dessas técnicas que você vai fazer para o paciente, fisioterápicas específicas, mas são fisioterápicas, né, aplicadas para o caso aqui da negligência, né? Então, é isso que é o nosso diferencial. A maioria dos trials, eles focam só na modulação. E eles até podem fazer algo, mas muito pouco, né?
A potência do aparelho é sempre a mesma?
Vai ser, no geral, isso está pré-definido. O ideal seria essa individualização, mas com o equipamento que a gente vai ter, a gente conversou com um colega, inclusive, da Austrália, ele falou que é bem difícil, porque, assim, com o parâmetro que a gente vai ter, vai ser... Por isso que a lesão em si é a mesma coisa, que você poderia questionar, ou alguém poderia questionar, about the size of the lesions, or the localization of the lesions. Cortical, subcortical lesions, very different situations. And these situations in stroke is very, very difficult. Homogeneous group of patients is very difficult. Impossible in general. Right. TDCS versus TBS, frequency. TDCS is 2 milliamperes during around 20 minutes. Localization of the 10-20 system, né? 10-20 system. And TBS is a technique concentrate fields in 3 pulses at 50 hertz, repeated at 5 hertz the frequency of frequency during around 3 minutes. It's very important, the protocol, physiotherapy protocol. And during the sessions, of the neuromodulation sessions, 3 times per week, during around 30 minutes, many exercises will be done using specific techniques, task, worked for these patients. In the specific case of the neglect, visual techniques will be used, visual techniques and the other techniques, sensorial techniques, used in this trial. Follow-up is a good question about this. How long time during the effect in clinical practice? This question do not answered in this case. Is still answered. 1 year, 6 months, 3 months, I don't know. These patients will be visited during the fifth, at the end of the sessions, and the session 15, and the 3 months, 6 months after 1 year or 12 months after the stimulation sessions ended. And this about the duration of the effect of the neuromodulation is a great question. It still remains the answer. Primary outcome is the beat, changes in the beat test. I hope better results in beat test for these patients. Beat test is like a very applied tasks, specific tasks, visual tasks in general, sensitive tasks too, and this punctuation is a cut of the punctuation is used for the included these patients and the observed in the end of the trial too. The secondary outcomes is very large. Is the control, the trunk control, frailty, about the quality of life, self-efficacy scales, fatigue scale, physical activity level, activities of daily living, and is very important in the topic, the secondary outcomes. Around the 18 participants per group, and it's a very difficult trial. Neurorehabilitation trials in general are very hard trials. The size of these trials is a phase 2 trial, as the phase of the safe trial, and this phase, and the sample is size estimated is small compared the other types of trials, mainly these industry trials, phase 2. However, it's very difficult. These patients was observed as very complicated patients. In many cases, this trial, unfortunately, don't bring good results or ended during this conduction phase. And I hope this trial bring good results and good perspectives, future perspectives, using the neuromodulation, non-invasive neuromodulation. The other aspect is the absence of the level of evidence in clinical practice in neurorehabilitation. Very less, very low evidence about this, and this when you make a trial, a neurorehabilitation trial, and in the end of this trial, you have a perspective, future perspective. We contribute for the future, future systematic revisions. In the future, the systematic revisions. In the future, this level of the evidence better. We will be better than today. As this, the CNPq FAPESP funding this trial, and this academic trial, this academic research, and this questions. Eu vou aproveitar e só fazer uma pergunta para o professor e para você, e para o professor Bafa também, em relação ao tempo, né, que você pesquisa lá, essa técnica, e se você tem algo de experiência lá, ou experimentalmente falando, ou viu alguma coisa em relação à prática clínica, mesmo para a parte psiquiátrica, para as questões de depressão e tudo mais, em relação à estimativa de tempo da duração do efeito. Se você consegue mais ou menos... Porque essa é uma pergunta que não tem resposta. Quanto tempo duram esses efeitos no ser humano após, por exemplo... Você pensa que no pós-AVC você tem a reorganização cortical, subcortical, você tem uma série de questões envolvidas, né? E você submeter esse indivíduo, né? Por exemplo, eu sei que se eu trato um paciente na unidade de AVC, ele vai ter benefício por até 10 anos depois da alta dele do hospital, porque isso já tem meta-análise, já tem o stroke trialists que mostram, né, essa situação. Mas em relação à neuromodulação, além de não ter evidência clínica para neuroreabilitação, alguns colegas têm até falado um pouco contrariamente, não sei qual o motivo, mas o uso, né, fora do campo da dor, ou fora do campo, por exemplo, para a neuroreabilitação, que é a área que eu trabalho, essa coisa me intriga em questão do tempo, porque é uma questão importante, né? Será que isso aí ele trabalha de uma forma, né? Vocês têm alguma ideia? Se você puder, por favor, perguntar para o professor também.
Okay. Yeah, Rodrigo is really innovating. Instead of answering questions, he's asking questions to you. So, Risto, he wants to know if you have, or Dubi, or I don't know, whatever in the audience, have any information about the lasting time of the effects. I mean, you do a neuromodulation. How long this will last in the subject? So, who is ready to comment and answer it?
Well, I think any effect, they last for a really long time, like we have memories. But it doesn't mean that the treatment effect, because the brain is very complicated, there are very many things going on, so that, for example, stroke therapy, this case maybe it doesn't necessarily last that long, because the brain is a homeostatic system. It tends to go towards some kind of preferred equilibrium. But something surely remains for a long time. That's my layman's answer.
But you don't know how long. It's a long time, but what it is long time? 6 months, 1 year?
Well, I think the point is that one should get the brain into a, not vicious cycle, what's the opposite? Into a self-healing mode, sustained mode, that's how the healthy brain works. It stays in the right mode. So if it's in the right loop, yeah, that will stay there for the whole rest of your life, I think.
You want to comment, Julio?
Yeah, I think that, it's very loud. I think the effects of RTMS or ITVS are about like 30 minutes to 60 minutes in healthy participants. And that evidence is basically from the motor cortex, because we see changes in the motor evoked potentials before and after. So it has been demonstrated that the effect lasts up to 60 minutes in the motor cortex. For different systems, we don't know, because different systems are different, right?
Okay, now questions from the audience.
Also wants to comment.
Carol?
sorry about my English, but in my clinical practice, I observed that the patients that have a better cortical reserve, the cortex more preserved, they have a better response and a long duration of the TMS and TBCS. So the patients have a good answer for 3 months without any section. Thank you. So in stroke and in negligence, we have protocols that we use 2 months of TMS and after one session for week. And the patients have a good answer for 2 months. And we start a new protocol with 20 days. So, and that's it.
So we still have time for some questions.
I have a quick question connected to what Julio was saying, that like that depends on the device and so on. So I saw that you have many groups that are going to run the study. So the instrumentation is the same for all? Are all of the groups using exactly the same setup?
Yes.
I don't know if I understood correctly about the position of the coil in the case of the TMS, but it's not clear for me. It's not related with the lesion. The position of the target area is related with the lesion or not? Any relation with that? Sem correlação nenhuma, nada?
A posição da bobina na lesão ou não?
Sim, na maioria dos casos, in the many cases, the majority of the cases, yes, it's the nearly of the lesion, the parietal cortex. The size of these lesions is very, very different. Cortical, subcortical lesions, lacunar lesions. The main priority is about the clinic, about the beat test. The beat test is the gold standard for the inclusion of these patients. The lesions presented only confirmed stroke, right hemisphere. The cortex parietal, left and right, simultaneously, the lesion, as possible, is near of the coil or the during session.
Okay, but you are not controlling by the size of the lesion. I know that it's very difficult to homogenize the patients.
No, the size of the lesion are studied after. Okay. The volumetry of these lesions, yes. Not control for the sessions. Okay. During sessions, no. After, for the good or bad results around the size or and the localization, yes. Or not guided for the stimulation.
Okay, thank you.
Thank you for your talk. I was curious about which kind of evaluation the patients are tested for.
Clinical evaluation?
Did you design any specific evaluation? For instance, we know that parietal cortex lesions can affect the verticality or,
yes,
I mean,
yes.
dissection tests. I was just curious to know which kind of tests you are applying.
You have many different types of. Catherine Berrego is on a scale created for the nursing in the past. Is a good scale, will be used in this case. I do not list it, all totality of these scales, many scales. And the beach test is the gold standard test around the world, and the verticality. I don't remember the pusher patients, the sign of pusher, né, is a major problem of the verticality after this type of stroke, of the neglect patients in advanced phase, this complicated situation. For these patients, the pusher, yes. As possibility, we identified these patients and the subgroups. Yes, more the specific tests of verticality. Our group described on sign of verticality in the neglect patients as the cross-leg sign. These patients cross-leg, cross-line, medial line, with a tentative of this find the space and in the neglect space, and this attention or the allospatial alterations is very interesting in this situation. The patient not recognized the left side of the world, and this part of the body and the rest of this is very, very difficult. And this very good. We have find a good results on the end of these trials. It's very promising, very, very good. For this patient specific group, 35% after strokes, a very common situation, clinical situation. We have a benefits for this group in the future using, for example, TDCS or TMS or both.
Any other questions? So I think if not, we should adjourn. We'll be here tomorrow at 9 o'clock. I think this first day has been very good. I learned a lot, and I hope to continue to learn. And I'd like to thank Rodrigo for his talk. I think it's very interesting to hear what people from the clinical side are demanding, because that's the ultimate goal, right? Help us to improve our life. And so with that, I would like to thank you and thank the speaker.
Vamos conversar.